Poter sedervi nelle file davanti se possibile, così riusciamo a stare più vicini, e aspettiamo giusto il coordinatore e poi cominciamo. Nadia, possiamo cominciare? So dear colleagues and students, it's a real pleasure for me to be here with you to start with the first event, first edition of the PhD Day. This event has been organized by the course of a PhD in biomolecular and health sciences at the Department of Biomolecular Sciences. And this event has been possible thanks to the enthusiasm and the hard work of specific persons that are here present, the Young Research Committee. The Young Research Committee is actually formed by the students that every year try to learn and provide energy and ideas for the organization of this event. And I asked the students just to stand up with when I call you. So Fabiana Fanelli, thank you, Fabiana. Veronica Gentilini, thanks, Veronica. Ludovica Montesi, that is probably just behind us. Alessandro Pennesi, Elena Perla, Lorenzo Scocchia, and also our representative, Pasquale Creanza from the PhD students, that is connected online, and all the PhD students involved in the organization that they had. Our teaching manager, Dr. Giulia Rivas, that is here present, thanks Giulia. The teaching committee that is led by Professor Lucertini, that is remotely following us today, and of course the event committee. Professor Frangipani, who experienced the PhD Day during her own PhD in Switzerland, and she was the one who bring here this idea, so thanks for promoting this event since the beginning. And the event committee. They are all here in present. So that is something important, very important. We want that the committee can really share and have the right impression from the presentations. And here let me to introduce to them. And Professor Andrea Pompa, Professor Fabrizio Perroni, Professor Mirko Fanelli. Mirko, I saw him before. Okay, here you are. Federica Semprucci, Eleonora Grassi, that is helping. Also Federica, and again Emanuela Frangipani, and myself, of course, Elena Barbieri. None of these would be possible without the solid support of our institution. So I would like to sincerely thank the counselor, or what is it, delegate, thanks, for research, Professor Giovanni Piersanti, and is here today to also considering future suggestions for the involvement of PhD students in the research organization for our department. And of course, with the generous funding and infinite patience of our coordinator, Professor Ferdinando Mannello. This event will not be possible. And now I am pleased to give the stage for the institutional greeting, so I ask to the delegate, Professor Giovanni Piersanti, for the research, and after that, the coordinator, and then I will leave the stage to Emanuela Frangipani, that she will take over as the moderator of the event of today. So let's enjoy this PhD day.
Hi to everyone, and I'm not director, of course, I'm here in spite of director, but it's really a pleasure to be here, finally welcoming you to this 4th edition of this PhD Day Uniworld dedicated to the research of our PhD students. Most of you, I think I remember from the class, so it's really a pleasure to see you finally here and do your short speech. Anyway, I was a PhD student as you 25 years ago, so it's really nice to see this big and nice manifestation and events. So, moreover, I'm really proud to see the quality and the diversity of the project and also the vitality of this doctoral program and the commitment of our young researcher, really. It's not only to share results, as pointed out there, it's really an exchange of ideas to strengthen the collaborations, to support the growth, and to make even more vibrant the academic community. I really enjoy this day. I wish you really fruitful day and fruitful collaboration and discussion. Enjoy the day.
So, thanks to all of you. A few words. I believe so much in my motto, and I hope that this day will be really, hopefully, an insightful moment and constructive moment for inspiring, for comparing, for exchanging the idea, the research of everyone. So I believe that also you will have a central role in these events because we have some different possibility to exchange idea. We have Samuele with us, and we are very happy for your presence now. And I try to find several projects and very high level, high profile of research. So best compliment and my compliment, personal and professional too. I believe that, of course, different fields of research and different opportunity to share, to inspire each other with different topics, with different research. So I would like to wish you an insightful and a fruitful meeting, leaving, of course, my personal motto. Never stop learning and learning every day and ever top learning. Enjoy.
Well, dear colleagues and students, I'm really happy to be here today, and I just wanted to say a few words. Today we come together not just to present research, but to connect across disciplines, to share insights, to explore what others are working on, to inspire, to be inspired. But I think that mostly we're here today because we have to remind ourselves that a PhD is not just a solitary journey, although it might seem like this sometimes. Behind every PhD thesis, there is a supportive community made out of peers, made out of supervisors, made out of family and friends that supports the students. And I think that days like today are really made to highlight this and especially to highlight the collaborative spirit that should be behind academic research to be successful. So we have a fantastic program ahead. We have an invited speaker today. We have presentations from second year, third year PhD students. We have poster presentations. We'll also have awards. So I encourage you to listen actively, to ask questions, and engage fully. Thank you very much for being here. I now hand you over to Veronica, who will introduce the speaker. Thank you, Veronica, please.
Good afternoon, everyone, and welcome to our PhD Day 2025 edition. It's a pleasure for me today to introduce our invited speaker, Dr. Samuele Negro. He currently works as a researcher in the University of Padova, where he also got his PhD title. His research is mainly focused on neurodegenerative diseases, including amyotrophic lateral sclerosis. When I was a master degree student, my Instagram algorithm made me run into one of his reels, and I would say that from that moment, me and my lab mates have become very fond of his adventures. Indeed, he's particularly known as some scientist through social media. So enjoy this session, and Samu, let us know more about you and your experience.
Thank you. And first of all, I want to thank all the organizers to invite me here. It's really, really, really a pleasure. I really like this kind of initiative, this kind of PhD days. I think that they are really important, and I hope that more universities are going to do this kind of initiative. So the title is a little bit changed from the sheet. So this is exactly what I'm currently doing. So as you can see, I'm studying ALS, amyotrophic lateral sclerosis. But today you will see a presentation about my research work, but connected to my activity on social media. So first of all, who I am. I'm a researcher at the University of Padova. I'm working here in this. I must admit that it's not beautiful at all, this structure. Now, after that, I saw this really beautiful campus. Congratulations, really. I hope that in future we can have something similar. But today, as I told you, I'm going to connect my research side with my social side. So some years ago, I create this page on Instagram with the name of some scientist, so the fusion between my name, Samuele, and scientist. But then you will see, I will tell you better how this page was found and why. But let's start from the boring part. I would say no, I'm kidding. Amyotrophic lateral sclerosis, ALS, is a relatively rare disease. You can see the incidence is 1 to 3 cases per 100,000 people. Is an age-related disease. You can see peaking around 60 and 80 years old. And so, as you can imagine, the incidence probably will increase due to the aging of the world population. And you can see the survival from the time of diagnosis is really poor, 3 to 5 years. Since today I'm talking about science and social media, I want to give you an example of how social media can be useful for research. I don't know if you remember the Ice Bucket Challenge. In 2014, there was this that now are called viral trend. So basically, people were recording themselves throwing an ice full of ice in above on their self. And this was done to try to raise money for research in ALS. And this viral trend really worked. You can see, I don't remember the name, but the number, probably $20 billion were raised. And this money helped to find new genes correlated to the disease and help to have the first trial, clinical trial, and the first therapy approved, Riluzole. And moreover, a lot of improvement in the clinical approaches to the patients have been done with this money. So, ALS is a neurodegenerative disease where basically there is degeneration of both upper and lower motor neurons, leading to complete muscle paralysis and eventually to motor neuron death, till the death of the patients. So as you can see, the pathogenic mechanisms are many. It is not known exactly which is the main of this pathogenic mechanism. As you can see, there are alterations in RNA metabolism, excitotoxicity, impairment of axonal transport. So many of them seems are involved in the disease. Moreover, there are sporadic forms of the disease that are 95%, and only 5% are familiar form. Many genes and several mutations have been associated to the disease. Here I reported the 4 major genes related to the disease. There are currently 2 theories about the onset of the disease. The dying forward hypothesis suggests that degeneration begins in the cortex, in the motor cortex, and then proceeds in an anterograde fashion to the cortical spinal projection. While the opposite theory implies that degeneration begins at the level of the neuromuscular junction, which is the anatomical structure that allows communication between motor neurons and muscle. And degeneration then goes back to the motor neuron soma in a dying back manner. And in our lab and many other groups strongly believe in the dying back hypothesis. So the one that is starting from the neuromuscular junction. And here I show you a picture, try to summarize what is the NMJ to help you to understand the presentation. Here you can see this cartoon where you can appreciate the motor axon terminal, the presynaptic Schwann cells, which are specialized glial cells that are involved in the maintenance and the plasticity of a neuromuscular junction and the muscle fiber. Here you can see a microscopy picture. In green, these are Schwann cells. In gray, we have the muscle fiber. In red, the motor axon terminal. So keep in mind this structure. Because as I told you before, this theory suggests that it is the NJ which is the early target of the disease. So the generation of motor neurons starts from the motor axon terminal at the neuromuscular junction. There are evidence of this in both in ALS mouse models, but more important in patients. And what is really important for my research, you will see later, is that before the definitive loss of the motor neuron muscle contact, there are continuous cycles of renal degeneration and regeneration events. So this means that in the early stages of the disease, there is a kind of plasticity of the neuromuscular junction that try to counteract the progressive degeneration. And therefore, the aim of my research is this one. I want to try to preserve, to stimulate, to boost this intrinsic neuromuscular junction plasticity to see if I can counteract the onset of the disease or the progression of the ALS. But, so the question is, that we ask to us is how we can preserve, boost this neuromuscular junction plasticity. And this story began when I was a PhD student. And I want to say that I would have loved to receive this kind of suggestion that currently I'm giving to the new PhD students. You will see many times me running.
Allora Giorgia,
Sei pronta ad iniziare questa avventura del dottorato? Adesso ti faccio fare un po' un giro di laboratorio, però partiamo da questa stanza, questa è fondamentale. Qui potrai venire a piangere quando ne avrai di bisogno. So you have to find, first of all, a room where you can cry during your PhD. So I cry a lot, but to summarize, during my PhD, it's really, I summarize a lot. We need 4 hours to arrive at this cartoon. So it seems easy, but it was really difficult. What we basically found using particular innovative tools that we have in our lab, such as neurotoxins that specifically target the neuromuscular junction and cause a degeneration which is followed by a complete regeneration of the structure, and also using other approaches that are the classical approach, which is the compression or cut of sciatic nerves. Basically, at the end, we found a signaling pathway which is involved in peripheral nerve regeneration, which is composed of by the chemokine, CXCL12. You can see here, this is a normal neuromuscular junction before intoxication. We give the neurotoxin, for instance, we have degeneration of the motor axon terminal. The green perisynaptic Schwann cells are activated, engulf nerve debris, and release the chemokine, CXCL12. Here you can see in white the chemokine, CXCL12, in perisynaptic Schwann cell, in green. What happens? The chemokine is released, and through the receptor, it's a specific receptor, CXCR4, which is not expressed in wild type, in control animal, in adult mice, is instead re-expressed upon a damage of the motor axon terminal. It is expressed in the motor axon stump. So what we found is that this pathway is really involved in PNS regeneration. And if we boost this pathway, we can have a faster regeneration. Instead, if we block this pathway using neutralizing antibody or antagonist against the receptor, we really delay regeneration after our experimental model approaches of damage. And how the pathway works, we found that basically the chemokine through the receptor is able to stimulate axonal elongation. As you can see here, we add the chemokine to primary motoneuron in vitro. You can see there is an increase of axon length. And here, using microfluidic devices, where we can plate the motoneurons in the somatic chambers and axons growth in these microgroove channels, we can add a stimulus in the distal chamber, creating a gradient. And here you can appreciate that the chemokine promotes axon growth. So we got a signaling pathway involved in PNS regeneration, and it does so promoting axon growth. So after this arrived a period of a fellowship. After a fellowship, assegno di ricerca, borsa di studio, not taxed borsa di studio, I was lucky. Now probably it will be a problem. But so I survived also this period, otherwise I would not be here. Okay. Thanks to my parents, I survived many fellowships. And in this period, basically, what I did together with the people in the lab, so the idea was to use the chemokine as a pro-regenerative drug. The problem was, first of all, it is really, really expensive to have a lot of them, a lot of the drug, and moreover, the clearance of the chemokine is really rapid. So we decided to design a synthesis of a new ligand of the receptor, which is called NUC390. So during my postdoc period, basically what I did is I tried to see if the ligand, this new drug, recapitulates completely the effect of the chemokine in all our experimental models. So we found that we have the same effect of the chemokine with both our neurotoxins model and also with the approach of the compression and cut of the sciatic nerve. So now in our hand, we have basically a pro-regenerative drug, and we can produce a lot of that. So now finally, we arrived in the currently project that currently, it started in 2019, we submitted 20 days ago. So it was a long story. And the question was, finally we have this pro-regenerative drug that is able to promote, to boost neuromuscular junction plasticity. Can we try to use this drug to maintain, to sustain, to promote the plasticity of the neuromuscular junction that, as I told you before, even in ALS, in the first stages of the disease, is present. So this is the main of my project. And luckily, after the postdoc period, there was a moment that probably I was almost quitting the academia, but thanks to my incredible, I would say, writing skills, I won a grant that you will see later.
Non, rien de rien. Non, je ne regrette rien. Ni le bien qu'on m'a fait, ni le mal, tout ça.
It's incredible. I won the grant thanks to the starting grant of the what was the name? Ricerca finalizzata of the Ministero della Salute. I can continue my research in the ALS field now. So what we did, we are using the SOD mice model, which is the most used one for ALS studies, because basically it's the only one that well recapitulates the clinical symptoms of humans. And as I told you, we are trying to stimulate with the drug, with NUC, the MJ plasticity and see whether we can delay the onset of ALS and delay the progression of the pathology. And so we are giving the drug, and this is the okay. No. Okay, I changed the slide probably. Okay, I changed the slide maybe this morning, sorry, I forgot that. And okay, so come back. This is the beautiful of the Diretta, I would say. So we are here. First of all, of course, we say, okay, we want to stimulate with the drug, but we must have the receptor. The receptor should be expressed in our ALS mouse model. And we checked that, and indeed is expressed. You can see these are all pictures of neuromuscular junction at different stages of the disease. These mouse models basically have a pre-symptomatic phase that is 1 month. After 2 months, it starts to have the first symptoms, so it's called the early symptomatic phase. Then we have the symptomatic, where the clinical symptoms are evident, and the late stage, the final stage, 4 months. And you can see here in red, we have stained the motor axon terminal, the neuromuscular junction, and in green, the receptor CXCR4. And you can see that is expressed throughout the disease progression. So we have our target, so we can really treat these mice with the drug NUC 390. This is the experimental workflow that we did. We basically inject the drug 3 times a week, IP, and we perform several assays, behavioral assay, motor function assay, and we collected tissue sample, do staining, immunofluorescence, and other kind of assay. I'm not going to show all the data, just some of them. In the lab, we are really expert about, as I told you, neuromuscular junction functionality assays. For instance, we do CMAP recording, where we can measure MJ function in the muscle through stimulation of sciatic nerve. And we have also set up a ventilation assay that allows us to measure pulmonary function. This is really important because in the late stage of the disease, also for humans, basically the patient died for respiratory failure. So have a drug that can improve MJ functionality of all the different muscles involved in respiration is really fundamental. And for this reason, now we also bought a new tool, a new instrument that will allow us to measure till 15 different parameters related to the pulmonary function. And so these are just a small part of all the experiments that we done, but maybe are the two most important, because what we found is that in both cases, the treatment with NUC ameliorates MJ functionality. Here is the CMAP analysis. You can see the column is higher. This means that neuromuscular junctions are working better. And this is the ventilation assay. This is, you can see, mice treated with vehicle. This is the drop of the respiratory function. You can see that in mice treated with the drug, the drop is less evident than the mice untreated. So then, of course, as I told you at the beginning, the final stage of the disease, in the final stage, we have the death of motor neuron in the spinal cord. So we also check, in collaboration with a group in Torino, the survival of motor neuron at the level of the spinal cord, and we found that NUC treatment increased the survival of the motor neurons. And of course, these are all results that we obtain in the ALS mouse model, the SOD one, but we hope to translate our discovery to humans. And for this reason, I got access to muscle biopsies from ALS patients, where we found the expression of the receptor in symptomatic stages of patients at the neuromuscular junction. And moreover, in collaboration with a group of Rome, we are also using iPSCs, from which we differentiate to motor neurons. And so we can have the motor neurons from the patients. And this is really important because we found the receptors, as you can see, is expressed also in these cells. And this is really important because this can help us. Do you remember at the beginning of the presentation I showed you that 95% of cases are from sporadic patients, and where there is not an association with the genes that we know are involved. And this is really important for this reason, because it means that probably the activation of the receptor is a response of the generation in all the different subtypes of ALS. So our treatment can be maybe used really in all the different background of genetic ALS patients. And so, as I told you, finally we submitted 20 days ago this paper, a long story from 2019, and it was also a long story drafting the paper, creating the figures. We started in January, and it was during this period that I had the idea of this video, and now I think you will understand why. Arise from the fact that for figure one, really for this paper, I got almost 40 version of the figure 1. Indeed, we are joking that probably we don't know which is which one we submitted. We choose randomly. We try one of them. So to conclude the scientific part, really briefly, I want to stress this concept because it seems easy here, just 3 pictures, but it was a long story starting in 2014. And really, 20 days ago, finally, we submitted this big paper about the effect of the drug in ALS. But I'm here also for this, right? So not for just for the scientific part, but for the social part. So really briefly, they asked me to tell something about how SAMU Scientist, why SAMU Scientist has been created. And everything starting from these 2 pictures. In 2017, I opened an account on Instagram. And because I said, I want to do something, right? I want to try to communicate something. I started with try to be a food blogger. Negative results, very few likes, 3 comments. I guess some of my friends insulting me, for sure. Then I said, okay, food blog is not working. Let's try with a travel blogger. It's not working at all. And so during a dinner with friends, one of my friends told me, but why are you not going to post what you do in the lab? Because my friend is not working in the scientific world, he's doing something else, right? But he was always curious about my research. He asked me, what you did with the mice today? I was a little bit afraid because he keep asking me about mice, but okay. But it was in that point that I decided, okay, maybe I can try to post something related to my work. And this was the first post. And I post this because, as you can see, I went to a conference with a poster of the wrong size. I needed the horizontal one, I arrived with the vertical one, and after this is now, I would say, a marchio di fabbrica on me, because after this, many other posters have been appended in different conferences with the wrong size. But you can see here, I have the same number of followers of the previous post, but I got tenfold more likes, 2 comments, still my friends insulting me, however. But this was the starting point. After this, I said, okay, I'm going to post about my research life. But I understood from this post that probably I have not to post the classic, just show what I'm doing in a classic way, maybe in a boring way, but I have to change the way how communicate. And I decided to use, in my opinion, one of the strongest communicative weapon that we have, which is irony. And this is how it started, with some meme. In 2018, Renzi was really amazing. It helped really a lot me to do a lot of different meme. It was basically a source of thousands of meme. And you can see I initiate really using the irony harm weapon. And here it is me try to, I don't remember, maybe praise some gods or try some yoga meditation before doing Western blot. But the really increase of the page arrived when Instagram had the possibility to post short video. This was the first one. this was the first one, and the numbers were impressive. I got really, I don't know, 4,000 or 5,000 followers at that point. But you see, I got 400,000 views, a lot of likes, shared, etc. And I said, okay, I want to go on with this kind of video and try to see in a funny way the life, our life, right? Our life as a young researcher or older researcher now. and so it's what I'm doing now. And no, you will see later this part. Now I'm just to show you just I don't remember, 3-4 videos and one meme about some new videos that I did recently. My last 15. These are the cells of my last 15 days, because I don't know, they keep dying continuously. So I don't know if they are killing themselves or what, but they are dying. And another video. I mean, I probably now here you have a new building, so try not to do this. But in our old structure, you can see it's terrible. We have walls that are painting terrible yellow. It's the worst place in the world to do video. Really, the light is terrible. And we have not a lot of space. We are really a lot of people. And this happens frequently, I have to admit.
C'è la luna, mezza mare, mamma mia, mi maradare. Figli mia, cutterare, mamma mia, pensaccia tu.
They told me that in cold room we cannot put students. Okay, I agree. And I mean, I think that this, I hope, but I think that this stuff are not going to change even in the future.
Ricordi quando eri povero?
Sì.
Sembra ieri, invece oggi.
Ma come?
Ma perché ricordarglielo, dai?
And I want to give you hope. I mean, you see, you can wait, but at the end, we will do it. I mean, it's almost close to the retirement, but it will be in a permanent position, 70 years old, perfect. So I show you some of videos, some of my memes, what I'm posting currently, and but I want to give you some pro of what I'm doing if someone of you want to start this kind of activities. It was something unexpected, I have to say, but what really helped me, the social media, is building network and get collaborations. Because, for instance, I put the motor neuron survivor in the spinal cord because we got that collaboration because one associate professor of the group followed me on social media. After that, have a look to PubMed. They said, okay, it's also a kind of a good research. They are doing something interesting. And so we started a collaboration. And it's happening frequently, this kind of stuff. So I'm really happy about this, and I want to continue also for this kind of reason. Also because I'm having collaboration with the companies that are helping me sometimes with some reagents, but it's easy also to communicate with other groups in Italy and change suggestions. So I think that, okay, it's a funny way, maybe it's a way to say that we are together in the same stessa barca, no? But it's really helpful, I would say, to use social media in general, not maybe Instagram as I'm doing, but think about X or LinkedIn, try to connect each other. And okay, the other stuff is really are important, engaging with people, I'm sharing my research, I'm trying to inspire the next generation of scientists. This is not working at all. They are inviting me many times at the high schools, but no one wants to become a scientist. So I hope that is not my fault, but cons, there are of course there are some cons. One is, it can happen sometimes, not taken seriously within academia. It's normal, I think. There are people that maybe can like what I'm doing. There are people that can say, what fucking is doing that, not doing research, is doing a video, is losing time, 1 minute of each day to do a video. Is this? Because this was always the question, time management. They ask me, but how many times do you imply to do that kind of video? Really 1 minute. I'm not smoking. I'm not drinking a lot of coffee. I'm not doing that kind of poses. My colleagues is smoking. Did probably 10 breaks a day to smoke. I did 1 break, 2 breaks, and in one of that I'm doing a video. This is the time that I'm spending to do video. Then maybe I'm lucky to have the idea, of course, but this is the time that I'm using to do this kind of videos. And you can see that I'm not spending a lot of time to create beautiful videos. This is another kind of job. It's not what I'm doing. And of course, when you post something on social media, you can have haters and negative comments. And a suggestion, if you are going to do this kind of communication on social media, tell to your PI. My department, everybody knows what I'm doing. My PI know from the beginning, and sometimes they are giving me some ideas. They say, somewhere you can do a video about this. Okay, thanks for the suggestion. But otherwise, I have the solution, if you are not going to tell to your PI, just in the case, do a video like this one. Yes. Sorry, the audio was removed by Instagram because for some copyright reasons. And so, at the end, I have no really suggestion if you want to communicate on social media. You have to try to be authentic and original. The fact that now it's full of different accounts that are communicating in this way. And of course, I want to thank the group in our lab because it was not just my work, and especially I have to say Professor Cesare Montecucco. It is my scientific mentor, and also is helping me to have idea for the videos. So thanks, Professor, for both of the two sides of my life now. And the different collaborators, some of them that I got, as I told you, thanks to social media, and you to invite me here. And if you have some question regarding both the scientific or the social part, I'm here. Thank you.
Okay, thank you, Samu, for your interesting presentation. Yes, we are in time. So if you have some questions, it's the time now.
Thank you, Lo, for this really funny and interesting presentation. I'm already your follower, so thank you a lot for your videos. Sorry to come back to your project, but I'm really interested in that, even if I'm not in that topic exactly. I had some question about the molecule, I mean nuke. I'm interested in the fact that you inject it thrice a week. This is because of the brief of life, and you never mentioned the concentration you used. So I'm quite interested in that because actually I'm suppose you have some problems with the solubility of the molecule. So I'm also interested in that, in the delivery system you want to use to deliver that kind of molecule. And last but not least, I'm curious also in another point. I'm wondering whether your approach is compatible with the, I don't know how to say, but the time of diagnosis. So thank you, really.
So about the time of diagnosis, this is a really good question. This is a problem because in the mice we start from the early presymptomatic stage where they start to have some symptoms. Is this something that at the moment we cannot do in humans because they diagnose? There is one problem: is the time of diagnosis. Usually, the average you need more than 1 year from probably which are the first symptoms in humans to have diagnosis. So basically, you know to have ALS when it's really too late, because often start with some cramps, for instance, with fatigue in just one arm, and you just think, okay, maybe I'm just tired today, so you are not going to a doctor for that. And so there is a lot of research trying to find, for instance, blood biomarker, to try to find a way to diagnose the pathology earliest as possible. And this is one point. The second point is that currently there are a new drug that has been approved, is tofersen, is with the ASO technology. It has been approved with some readout as neurofilaments, okay. And but the fact that in that case is working for this familiar form of the disease, the SOD1, and in that case they are following the familiar and try to do the therapy before symptoms. So they in the early symptomatic phase to prevent the disease. And in that case, disease could be another option. So it's just for a small part of all the patients, but this is an option, right? Try to treat this kind of patient with ASO and maybe with another drug that can support the plasticity of the neuromuscular junction. The other question, is still really complicated. I mean, we have no problem of stability. The drug is stable till 40 days in aqueous buffer, even in acidic buffer, because the idea was also to try to do something, an oral administration. The clearance is really rapid. That's the problem. And indeed, now what we are applying for, hoping to have money, is first of all, we want to do a new high throughput screening to try to find new ligands. The second one is the approach that I'm applying for, is use adenoviral therapy. So use gene therapy to try to overexpress the chemokine in Schwann cells. Because I hope that you have not the same idea, because you are saying, okay. Because this is the idea. Because Schwann cells are the cells that are close to the motor neurons, and they sense the motor neurons that is degenerating, and they respond to the degeneration. And it seems that in ALS, the Schwann cells are not able to respond to the disease, or they lose this capability during disease progression. And indeed, they produce the chemokine in the early stages of the disease, but not in the symptomatic or late disease stage. They are not able to produce the chemokine. So I want to overexpress the chemokine. And this will help, because in this way I will have the overexpression of the chemokine really close to the motor neuron, and I will avoid all the possible systematic side effect of a drug that is involved in a signaling pathway that, however, is involving many other, for instance, inflammatory approach mechanism or metastasis, for instance. So this is the follow-up of the project. Just to conclude, the importance of the work is, okay, the drug, but is also the biological messages that we are giving, is that stimulating, boost the plasticity of the neuromuscular junction can be a therapeutic approach. So now in the lab we want to try some way to do that. So with the chemokine, or we have other drug or molecules that we are testing.
Okay, thank you, Samu, for being here with us, for your availability. It was a pleasure for all of us, I would say. And we have a little gift for you here. Okay?
You can promote our doctoral course. No, well, now it's a bit, yeah, okay. Too hot today, but thank you very much for being with us today. We will all be rooting for you and keep our fingers crossed for your publication that you just submitted. Maybe later you will share with us which journal you've chosen for this 7 years publication. Awaiting. Okay, so let's move on to the PhD student session. We will start from the second year PhD students and then move on to the third in alphabetic order. The only change that we are going to make is that for the students who are actually working abroad during their secondment, they will be all grouped at the end of each session, okay? So now I hand you over to Lorenzo and again Veronica, who will moderate this session.
So, the first one, Bargagni Eric.
Okay, welcome everybody. My work is on the role of nitrosative stress and endoplasmic reticulum stress in oxysterol-induced endothelial dysfunction. Endothelial dysfunction is a pathological condition characterized by disruption of vascular homeostasis and a pro-inflammatory state. Endothelial dysfunction has been identified as a critical early step in development of arteriosclerosis. OxLDL and oxysterol have been identified inside the arteriosclerotic plaques, and this suggests us that they have an involvement in the development of endothelial dysfunction. So my work focuses on a specific oxysterol, secosterol B, and its effect on the UVEC cells. During the first year of my PhD, I already demonstrated that secosterol B is able to induce nitrosative stress with increased level of nitrous oxide and reactive nitrogen species, and a depletion of glutathione. Moreover, secosterol B is also able to induce endoplasmic reticulum stress with alteration of the membrane, activation of the unfolding protein response, and accumulation of misfolded proteins. During the second year, I focused on protein S nitrosylation. This is a post-translational modification of protein in which a nitrous oxide group is bound to a protein, a cysteine thioylon of a protein, and this can affect the function of the protein itself. We were able to demonstrate that secosterol B is able to increase protein S nitrosylation, and with proteomic analysis, we know that this is due to 39 proteins that are exclusively nitrosylated in secosterol B treated cells, and 18 proteins that are more nitrosylated in secosterol B treated cells. Among these proteins, we found PDA6 that is exclusively nitrosylated and GRP78 that is more nitrosylated. These are both proteins involved in protein folding and in the unfolding protein response. So to test this, we used an inhibitor of nitric oxide. This should reduce protein nitrosylation, and we want to see the effect on the endoplasmic reticulum. So testing with LNAME, this inhibitor of nitric oxide, we can see that we have a reduction in the alteration of the endoplasmic reticulum, and also we have way less protein aggregates in these cells. Looking at the distribution of GRP78 in the isopycnic sucrose gradient, we can see that LNAME is able to reduce the formation of these protein bodies. Moreover, pretreatment with LNAME is also able to reduce oxidation of PDI, another protein involved in protein folding. So in conclusion, we can say that sequestrol B induces nitrosative stress and endoplasmic reticulum stress, can promote protein nitrosylation, specifically of GRP78 and PDA6, and moreover, inhibition of nitric oxide can alleviate both endoplasmic reticulum stress and the accumulation of misfolded protein. I want to thank everybody, and special thanks goes to my supervisor and co-supervisor.
So thank you so much, and there is a space for one question. If someone? Everything clear? So, thank you.
The next student is Cherubini Alessandro.
Questo in senso. Questo è il puntatore? No? Vabbè. Evito che punta. No. Okay, this one. Okay. Good afternoon, everyone. It is a great pleasure for me to be here and to introduce you my PhD project, which focuses on the validation of a new pharmacological treatment for breast cancer. Our strategy is the so-called targeted therapy, which consists in the identification of a specific target whose inhibition may blunt cancer aggressiveness. And the target that we are interested in is the endoplasmic reticulum oxidase 1, ERO1, which is responsible for the disulfide bond formation in newly synthesized proteins. I can't use this pointer, sorry. And but why ERO1 may be an important target for cancer treatment? Because it is upregulated. It's upregulated by the unfolded protein response, an homeostatic response that reestablishes the balance between the synthesized and the folded proteins. And is upregulated by apoxia, which is a very common condition to most solid cancers. Thereby, ERO1 plays a crucial role for cancer fitness because it improves proteostasis and it improves the secretion of many factors, and among them the VEGF, the vascular endothelial growth factor, the master regulator of angiogenesis. So the project consists in 3 tasks. The first one has been the synthesis of new analogs, inhibitors of ERO1, starting from EN460, which is an inhibitor suffering from poor water solubility and off-target effects. And then the inhibitors have been tested in vitro in cellular model and then characterized in vivo in animal model. The first task consists in the changing of the main pharmacophoric groups of this inhibitor. I just want to highlight the green one, which is the electrophilic species, which acts as a Michael acceptor and binds covalently to a cysteine residue in ERO1. This is the docking site analysis, and you can see the yellow one, which is the Michael acceptor and binds to the cysteine, and that inhibits ERO1 in a reduced and inactive state. Then all the inhibitors have been screened, and the ones with the lower IC50 have been selected for the in vitro characterization. Here we can see non-reducing Western blot, and I have highlighted with the arrows the ERO1 in a reduced state, which is the inactive one, and then the inhibitors have been assessed for the cytotoxic effect, and you can see that there is a dose-dependent and also a target-dependent cytotoxic effect of the inhibitors, comparing the wild type versus the ERO1 knockout murine breast cancer cells. And this is confirmed also by the cytofluorimetry analysis. You can see that there is a dose-dependent apoptotic population that increases the levels, you can see. And this result, the inhibition of ERO1, resulted in an impairment of the migration potential of cells, of the cancer cells, which is also dose-dependent. So, we have seen that the inhibition of ERO1 is dependent on the presence, and that the I2 impairs cell migration more efficiently at the EN460, and that EN460 gives rise to more apoptosis than I2. The last part was the in vivo characterization in tumor-bearing mice, and we have inoculated breast cancer cells in syngeneic models, and we follow the tumor growth and by daily caliper measurement, and what we have observed is that there was a significant difference in terms of tumor growth rate comparing the treated versus the untreated versus the controlled. And the inhibition also resulted in an impairment of the VEGF secreted by these cells. This is a ex vivo analysis of the tumor homogenates, and by VEGF ELISA we have measured the levels of VEGF. And also we have studied the tumor microenvironment by the orthotopic implantation in the mammary fat pad, and we have observed that there is an impairment of the PDL1 expressed by the myelo-derived suppressor cells. So, to conclude, we have seen that ERO1 inhibition by a small molecule is feasible, that this inhibition gives rise to a cytotoxic effect, and that this is impairment of VEGF secretion and migration. And very interestingly, we have demonstrated that the inhibition of ERO1 impairs PDL1, which is an immune checkpoint, is a mechanism by which cancer cells elude the immune system surveillance. And so this gives rise to further analysis. And I thank all my collaborators, and of course, I thank you all for your attention.
Are there any questions for Alessandro? No? Okay. Thank you. The next one is Ciurlia Eugenia.
Good afternoon, I am Eugenia Ciurlia, and my project aims to set up a methylation-sensitive high-resolution melting assay as a non-invasive approach for the early detection of cancer. In particular, starting from liquid biopsy, we analyze the cell-free DNA, which is a mixture of small DNA fragments normally released after cell death, whose levels in cancer patients are increased due to the presence of circulating tumor DNA released by cancer cells carrying all genetic and epigenetic aberrations. So this assay starts with the purification of cell-free DNA from blood, its conversion with the sodium bisulfite, and the amplification by quantitative PCR followed by high-resolution melting analysis as a fast and cheap method for studying methylation. We designed 3 primer pairs and selected the largest one, capable of amplifying the human pericentromeric satellite 2 DNA, which is among the best characterized hypomethylated sequences in different types of solid tumor. First, we tested the assay on cell-free DNA samples from healthy donors, as well as on genomic DNA from tumor and normal cell line, and also on standard DNA, completely or incompletely methylated. Successfully, the assay reveals the different methylation pattern between normal and tumor DNA, as shown by 2 different methylation curves. Then we test the sensitivity of the assay in detecting tumor DNA when present at different concentrations and different methylation levels in a normal background. To recreate these conditions in vitro, we mixed known concentrations of demethylated DNA derived from tumor cell line with the normally methylated cell-free DNA samples. The results reveal a significant dissociation curve when the tumor DNA was equal or greater than 10%. In order to optimize the sensitivity, we modified the thermal profile with the cold PCR strategy. consisting in a first PCR with the normal thermal profile, followed by a second amplification of PCR product with a lower denaturation temperature in order to specifically amplify the demethylated DNA, thereby enhancing the ability to detect tumor DNA up to 2% of the total amount of normal DNA. At the moment, we are testing this assay on samples from cancer patients in different types and stages of solid tumors. I want to thank my labs and thank you for the attention.
Thank you. Any questions? Okay. Move on with the next one. Essa Manar Magdi Labib Mohamed.
Good afternoon. My name is Manar Asta. Today I will present a brief overview of my research projects conducted under supervision of Professor Annalisa Luigi and Professor Michele Guscini. Right now I'm working on 4 projects under a common theme, which is development of nanoparticle to improve the therapeutic delivery, drug bioavailability, and targeting efficiency. These nanoparticles are, I'm sensing them by microfluidic technique. Microfluidic platform is enabling technology, enable me to have a precise control of the fluid dynamics and control the mixing of the ingredients. This will result in fine-tuning of particle size and polydispersity index and scalable with consistency batch quality. The first project is about creating and design PLGA nanoparticle loaded with rifampicin to improve the antimicrobial efficacy of the rifampicin and help in the meningitis treatment, going through the in vitro testing and safety and cytotoxicity assay through using the Galleria melonelli. The optimization have been done using design of experiments and then characterization like TEM, FTIR, in vitro drug release with a very promising results. And then we decide to conduct a cytotoxicity assay to check the survival rate and larvae dissections, which prove the safety of the formulations. For the project number 2, we conduct lignin nanoparticle loaded with curcumin in order to increase the curcumin stability bioavailability and to help in cancer treatment using microfluidic technique. The optimization have been done using design of experiments which show the optimized formula and validated using TEM. Further characterization are still in progress. Third project is about peptide conjugation or decoration for the PLGE nanoparticle to improve this nanoparticle to have more selectivity to specific cells and tissue. Actually, there is 3 attachment strategies: loading drug synthesis and absorption on the surface after synthesis during the dialysis, and the chemical conjugation. Now we are trying to discover the best attachment strategy that give us the best binding affinity and keep and maintain the nanoparticle stability. The last project is still in the preliminary phase. The objective is to improve the photosensitizer delivery to enhance the photodynamic therapy. By microfluidic technique, we are preparing PLGE nanoparticle to encapsulate the photodynamic, the photosensitizer drug inside it, and then integrate it in a hydrogel, and then to be printed to help in the drug delivery, tissue engineering, and hydrogel application. Thank you for your attention.
You have any questions? Okay, thank you. We can go to the next, Fanelli Fabiana.
Good afternoon, everyone. I'm Fabiana Fanelli. My supervisor is Professor Giosuè Annibali. Okay. The title of my project is WPS13B Deficiency Disrupt Golgi Architecture Without Compromising Classical Glycosylation Markers in Fibroblasts from Cohen syndrome patients. WPS13B is a giant peripheral membrane protein and is localized in Golgi membrane and is associated with Golgi architecture and stability. WPS13B mutation cause a Cohen syndrome. Okay. WPS13B mutation causes Cohen syndrome, and Cohen syndrome is a rare autosomal disorder characterized by many clinical features, including truncal obesity, microencephaly, and so on. There's a problem because less or dysfunction of WPS13B leads to Golgi apparatus abnormalities, including cistern fragmentation and swelling. There are limited informations about the impact of WPS13B mutation on canonical Golgi apparatus functions. So in our study, we utilized the fibroblasts from Cohen syndrome patients in compared to control fibroblasts to analyze glycosylation patterns in the classical glycosylated EVLI proteins like LAMP1, LAMP2, IGF-1 receptor, and IGF-1 EA. So our first Cohen patient is characterized by a long deletion involving exon 18-25, and the second and third patients are characterized by heterozygous deletion of exon 17 and heterozygous variants in exon 59. As expected, Golgi apparatus fragmentation was observed in Cohen syndrome fibroblasts. WPS13B exhibited a partial localization with the cis-Golgi marked, although with a reduction staining and border cytoplasmatic distribution in Cohen syndrome fibroblast. And in Western blot analysis, we have no glycosylation defects in the general glycosylation proteins, but there was an increase of molecular weight in LAMP1 and IGF-1 receptor, but also an increase of IGF-1 secretion in Cohen syndrome fibroblasts compared to the control. In conclusion, a gap in the study of this pathology is the possibility to identify and to diagnose this pathology under 5 years of age in Cohen syndrome patients. So our study suggests that the conventional glycosylation markers not constitute a reliable diagnostic tool for Cohen syndrome patients. And for this reason, in collaboration with CNR of Catania, we are developing a mass spectrometry to identify the glycoprofile in these patients. And we are also developing a CRISPR-Cas9 system to knock out WPS13B gene to clarify also the role of WPS13B in protein glycosylation. Thank you all for the attention, and stay more tuned for updates in my PhD project.
Any questions? Okay. So next PhD student, Gnoni Gloria.
Good afternoon, everybody. I'm Gloria, and my supervisor is Professor Stefanetti. So my project is entitled Immune Response Elicited in Animals by Carbohydrate-Based Vaccines Obtained Through Different Technologies. So I mainly focus on Klebsiella pneumoniae, which is a gram-negative bacterium and also the leading cause of neonatal sepsis in low and middle-income countries. but no vaccine are licensed against this bacterium, which displays two main polysaccharidic antigen in its surface, the capsular polysaccharide or K antigen and the O antigen. So I use the K antigen to obtain different vaccine candidates, classical glycoconjugate, but also MAPS technology, which is an innovative system based on the high affinity and non-covalent interaction between biotin and rizavidin, but also nanoparticle, which have an ideal size for antigen presentation. So for glycoconjugate, I use the 8-mutant tetanus toxin and the CRIM, so the diphtheria toxin, as carrier proteins. Here we have the generation of MAP system and the nanoparticles. So moving to results, all the platform have been characterized in size, molecular weight, and also polysaccharide to protein weight-to-weight ratio. And they were used to immunize mice and rabbits at day 0, at day 28, collecting the sera at day 27, day 42. So in rabbit, we can see high IgG titers elicited by all the platform, which CRIM conjugate that gave the lowest response. And this result was confirmed also in SBA, so in serum bactericidal assay. And here we have the IgG to IgM ratio. So we can see the switching from IgM to IgG going from day 27 to day 42 in both conjugate and nanoparticles. More or less, we obtain the same results also in mice. Again, we can see high IgG titers with the lowest response elicited by MAPS. Results again confirmed in SBA. And here we can see a statistical significant class switching from IgM to IgG in CRIM conjugate and MAPS. Here in mice, we can also look at different IgG subclasses. So thank you for the attention, and a special thanks to my supervisors.
Any questions for Gloria? Yes.
I use one type of K antigen of the pathogen.
Thank you. The next one is Iqbal Muhammad Yuanid.
Hello, good afternoon, everyone. My name is Muhammad Junaid Iqbal, and my topic is basically therapeutic potential of ATM variants in ataxia telangiectasia. My supervisor is Professor Michele Menota, and my co-supervisor is Anastasia Ricci. So basically, ataxia is a rare autosomal disorder which is characterized by progressive neurological decline, and even it can lead to different cancers such as leukemia and lymphoma. So it is a structure of ATM. Okay, so basically it is present in inactive form. When it gets active, under the stress of DNA damage, it is active as a monomer, and at serine residue it got phosphorylated. And when it gets active under oxidative stress, it is activated as a dimer. So basically the role of ATM protein is it is active for the DNA repair, for homologous repair, regulating cell cycle, and also managing oxidative stress. So in our previous studies, we have seen that when we give dexamethasone treatment to ataxia patients, we have seen that various splice variants were produced which were helping to regain the function of ATM in ataxia telangiectasia patients. Two of those variants were very promising. One was the natural variant ATM453, while the other was ATM-SINT, which was computationally designed by Anastasia in our lab. And we also have a patent for that. So our main objective is to check that if these variants, okay, these are helping regain function, but how they are doing that. This is our first objective. So we want to see whether these variants are forming a dimer with endogenous ATM or not. And secondly, we also want to see whether these variants are interacting with some other proteins or not. So we can use it as gene delivery application. So as you say that we have done a Western blot, we have basically introduced through lentiviral system these variants into human fibroblast cell lines, and then we observed through Western blot. As well as we used biotinylation and avidin beads capture to see that if our variants are interacting with other proteins or not. So basically with our results, we have successfully seen that our variant is forming dimer in all the cell lines under oxidative stress, 453 as well as Sint. So this was the first thing that, okay, our dimer is regaining the function through this dimerization, hopefully. And secondly, we have seen the biotinylation experiment. It is in the first phase right now. So we have seen that, yes, our variants, this is our confirmation of our variant present in the cells. So our variant is interacting with many different proteins, and we are going to now do mass spectrometry analysis to see which proteins are interacting with it. So this is mass spectrometry analysis, and we have seen that our variant is attaching in the same side of endogenous ATM when two ATMs join. And other than that, I have also worked on a different kind of study. It is a retrospective analysis, research on overall hereditary ataxia, all type of ataxias. It is done on US CDC Wonder data. So this is just a side thing that I want to explore, that how in all type of ataxias the mortality is increasing with time. So we have seen that mortality in male is increased as compared to females, and in whites mortality is more as compared to African Americans, and as well after 2015 the mortality is increasing very highly. So there must be some treatments, because hereditary ataxias are rare diseases, so there is no treatment right now present. So at the end, we have seen the dimer formation, and at the end, we have seen that various proteins are interacting with that. So in future, we want to use some multiomics analysis through NGS, transcriptomics and proteomics, to see how our variants rescue, especially in the scene of non-coding RNAs, microRNA, long non-coding RNA. So this is in future I will do in the next year. And at the end, I want to thank my professor, Michele, who is right now not here, and also my co-supervisor, Anastasia, and all my lab colleagues who were always very supportive, especially in understanding my English. Thank you so much.
Thank you. Any questions? Okay, thank you. Next one, Perla Elena.
Good afternoon, everyone. I'm Elena, and as most of you already know, I'm working on RBC-EVs as a novel RNA delivery system. Okay. Thanks to their unique properties, RBCs are already used as drug delivery system into clinics, thanks to the use of the red cell loader. And as almost all other cell types, RBCs can release extracellular vesicles that are involved in cell-to-cell communication. But the advantages of using RBC-EVs as drug delivery system include the easy accessibility from their source cells, from donors or patients, the lack of DNA, the high biocompatibility, and the possibility to be long-term stored. So we decided to produce our RBC-EVs starting from preloaded RBCs. So RBCs underwent epitonic dialysis for the cargo loading, thus obtaining preloaded RBCs that were then induced to vesiculate by our newly patented soft extrusion method that resembles the squeezing of RBCs into the spleen. Then the loaded RBC-EVs were deeply characterized according to the minimal informational study to assess that you are actually working with extracellular vesicles. Recently, I've been working on this project for the GAMT mRNA delivery using RBC-EVs. And for what concerned the loading efficiency, we had to face many issues, but, sorry, some spoilers. I was saying, from the loading efficiency point of view, we had to face many issues, but at the moment we are able to encapsulate this long RNA into RBCs for the first time, that is also retained into RBCEVs with very high successful rates. Then we moved to pharmacokinetic studies, and our RBCEVs demonstrated a longer than expected half-life into the circulation, with an accumulation primarily into the liver and no significant accumulation into the spleen. Moreover, the hepatocyte uptake was assessed using immunofluorescence. Finally, we evaluate also the therapeutic efficacy. And as you can see, a single administration of our loaded RBCEVs decreased the guanidinoacetate levels in blood. So, to summarize, we were able to optimize the long RNA loading procedure into RBCs for the first time with the production of, I don't know, doesn't matter, with the production of RBCVs with great properties and very high loading efficiency rates. But most importantly, our RBCV-based treatment is not only feasible, but also efficient. So to conclude, stay updated for further results, and thank you for your attention.
Thank you, Elena, for your presentation. Any questions or curiosities? No? Okay. Thank you. The next one is Sartini Alice.
Hello, everybody. I'm Alice Sartini, and this is my PhD project on how to develop a carbon-engineered organs on a chip for brain injury repair. A lot of my project revolves around the neurovascular unit, which you can see here is composed by a lot of different cells, endothelial cells, neurons, astrocytes, and pericytes. And this structure is crucial for brain homeostasis. And in fact, its dysfunction has been associated with many different central nervous system disorders, such as traumatic brain injury, which as of now has no effective treatment. So what we propose to better study this structure and this impairment is to recapitulate it inside an organs on a chip device, which allows us to co-culture all the different cells together in a controlled environment, which is a lot closer to physiological condition when compared to classical in vitro cultures. And what we want to do is add carbon-based materials, which are very small in dimension, they're conductive, they're resistant, and have shown to be able to modulate neuronal activity, which is very interesting for this type of dysfunction. So what we have chosen to use are carbon nanotubes, which I use as a substrate for primary neuronal cultures. This way I can assess their effect on neuronal activity by doing patch clamp recordings. And by doing so, I can see the cell's electrical activity. And just an example of what I see while I record, these are all single synapses that one cell can do. And one main effect of these nanotubes is an increased synaptic activity, not really in frequency, but rather in number of cells that are able to have synapses. Next, I wanted to do some calcium imaging on the cells, and you can see here they are all lining up together, meaning that these cells are mature neurons, and they were able to form a complete network. So I have seen their functionality. Next, I wanted to see their morphology, and I wanted to see how they interact with the nanotubes. So on the left, maybe not very well, but you can see some immunofluorescence studies. And on the right, on the top, these are neuronal dendrites and axons. And on the bottom, these are the nanotube mesh. Parallelly, we developed our organs on a chip. So we designed it, we 3D printed it, and you can see here there are different layers in which we culture all the different cells to make the neurovascular unit. Finally, we wanted to make the model as human relevant as possible. So what we did was starting differentiating iPSC-derived cells with rather neurons. And as of now, they do show the markers for neurons, for astrocytes, and they are able to generate action potential when stimulated. But the goal here will be to have spontaneous functional activity. So the future step will be to put all of it together, so to have the chip with our carbon nanotubes and our cells and see the effect. So I want to thank you all for your attention and my group for the big help.
Thank you. And any questions? Okay, thank you so much. Then move on with the online students, and the first one is Pasquale Creanza.
Hello. I share my desktop. So, can you see my presentation?
Yes.
Okay. So, good afternoon, everybody. I'm Pasquale, and I'm a second-year PhD student. My PhD project regards a biotechnological approach for the production of new eco-friendly bioplastic. So, petroleum-based materials, thanks to their easy processability and low cost, have acquired more importance in our lives. However, the wide use of these plastic materials is proved to be very problematic for the ecosystem, representing today one of the major sources of pollution. To overcome this problem, the main goal is trying to replace traditional polymers with plant-based biopolymers. The idea is to use an edible protein called fasolin that represents the 50% of vascular storage protein in common bean seeds. However, the native fasolin is organized in homotrimer and are unable to form these long polymeric chains. Thanks to the genetic engineering techniques, it was possible the insertion of a cysteine before the 4 C-terminal amino acid sequence of the protein. The presence of this cysteine can bring the production of disulfide bridges between the single fasolin units, and this can lead the production of a long polymeric chain of the proteins. So the working hypothesis is to use this, is to engineer the native fasolin with the insertion of a cysteine, then transform the chloroplast of Nicotiana tobacco plants in order to have transplastomic plants. And once the biopolymer is produced and purified, it can be subjected to a plasticization procedure to obtain different plastic materials for ranging from packaging to biomedical approach. So, to verify the presence of the biopolymer, an analysis on the level of the aggregates was then carried out by comparing the native fasolin with the engineered fasolin, and we can observe that in the native one there is only the presence of the monomer, while in the engineered one we have the monomer, the dimer, and also the presence of the aggregates. And these let us know that the cysteine engineering strategy works, allowing the production of a high molecular weight aggregates. Then the biopolymers was subjected to electron microscopy analysis, and we can observe its typical fibrous structure that is essential for the plasticization, how the characteristic of the plastic products depends on how these units are arranged in space. So after the purification, the biopolymer is subjected to plasticization tests, and we can observe that it can form thin films that are transparent, insoluble, and elastic, even if no other tests need to be carried out to verify the chemical and mechanical properties of this bioplastic. So the ongoing of this project is the development of new protocols to simplify the extraction and the purification of the biopolymer in order to reduce production costs and increase the yield, the transformation of plants used for crops production, so we can use the waste that they generate to make the biopolymer, making a circular economy model where waste become a resource. So we would also like to modify the structure of the homotrimers to make more resistant bioplastic, and also develop a strategy for the cell culture that could be used for the production of the bioreactor and also for the production of the bioplastic. So I want to thank my supervisor, Prof. Andrea Pompa, and my lab team, the Prof. Michele Osnato and Elisa Maricchiolo, and all of you for your kind attention.
Thank you, Pasquale. Any questions from the audience? No? Okay. Thank you.
Okay. Thank you.
The next one is Giolitto Chiara.
Yes, good afternoon.
Can you share your screen, Chiara? Okay, let's move to the next one. The next one is Morganti.
I'm sorry.
Ah, sorry.
It logged me out. I don't know why.
Okay.
I tried to share my presentation.
Yes. Okay.
Can you see the presentation?
Yes.
Can you hear me?
Yes, we hear you.
Okay. Okay. So good afternoon again to everyone. I'm Chiara Giolitto. My supervisor is Professor Panelli Mirko, my co-supervisor Amatori Stefano. And the title of my presentation is Epigenetic Therapy for APL, Study of New Epigenetic Therapeutic Strategies. My PhD project starts from an experimental hypothesis under patent, which supports the possibility of seeing synergy in combined treatments between a molecule named maltonis and other epigenetic approaches. Among other effects, maltonis was demonstrated to induce a marked reduction of histone 3 H3 lysine 9 trimethylation, a translational modification known to be involved in the regulation of a broad range of biological processes, including the establishment and maintenance of specific DNA methylation patterns. Considering the known crosstalk between histone modifications and DNA methylation, it was worth wondering the possibility of making maltonis work with other non-epigenetic approaches like decitabine, well known for its DNA demethylating activity, and if their combination could lead to a synergistic effect. For this reason, in the first part of my PhD period, I evaluated the biological effects of maltonis and decitabine alone or in combination in time course and dose response experiments up to 72 hours of treatment. But unfortunately, despite their biological activity, combined treatments gave contrasting results. In fact, as it can be seen from the combination index used by the Tutaralai method to define the presence of or absence of synergy between two molecules, at 72 hours of treatment, synergy doesn't appear to be as credible as I initially hypothesized. The working hypothesis was that they could synergize because of the epigenetic marker H3K9 trimethylation, which should decrease as a function of both decitabine and maltonis treatments supported by Litri-Cha. On the contrary, as shown by Western blot analysis, the combinations which gave indication of synergy don't actually downregulate this histone marker, supporting the hypothesis that synergy is not so robust. Nevertheless, the decitabine treatments returned an unexpected data. In fact, the decitabine-induced increase in H3K9 trimethylation in a dose-dependent manner, presumably due to an attempt of cells to restore DNA methylation, is not supported by literature. So it's worthy of attention and further investigation in the coming month. In the meanwhile, thanks to the collaboration with the European Institute of Oncology, we had the possibility to set up an in vivo pilot experiment in NSG mice, aiming to observe an anti-tumor effect in therapeutic combination. The experimental scheme for the mice involved subcutaneous inoculations of NB4 cells, 1 million cells per inoculum, and the start of treatments 24-hour after cell inoculation, with the division of mice into 4 groups. Maltonis, those treated with Maltonis, 20 milligrams per kilo, those treated with Olidac, 0.4 milligrams per kilo, those treated with the combination, and those treated only with PBS as control. As you can see from the weight of tumor collected at the end of the experiment, unfortunately, this attempt don't provide data on synergy, but shows again a very interesting effect of the decitabine alone, worthy of further mechanistic investigation as well. To conclude, this first set of data provides an initial indication of the direction of future experiments. As regarding in vitro experiments, the variability of synergy data highlights the need to refine the experimental procedures. And moreover, the modulation of histone H3K9 trimethylation, initially postulated as the mechanistic driver of synergy, seems not to be involved in the biological effect. However, the decitabine-induced increase in trimethylation levels of this repressive histone mark is an unexpected data, not supported by literature, that is worthy of attention and that I will for sure focus on in the coming month. As regarding in vivo experiments, given the lack of effect of Maltonis in vivo, we considered testing it in vitro at higher serum doses, hypothesizing an interference of serum itself with Maltonis activity. For this reason, I'm conducting in vitro experiments on NB4 cells in the presence of 50% serum, which actually show an inhibitory effect of serum on Maltonis activity, but data are not shown here. This makes it mandatory to find a delivery strategy to protect Maltonis from possible binding to serum proteins, which is typical for small molecules. In addition, considering the growing interest in including the methylating agents as a therapeutic strategy for the treatment of various types of cancer, and given the effect obtained by the decitabine in vivo, an interesting continuation of the project would also be to further investigate how this methylating agent works in vivo. So hoping to show you more results soon. I thank you for your attention, and I also want to thank my research team.
Okay, thank you. Any questions? Okay, thank you so much.
Thank you.
So the next one, Gianluca Morganti.
Good morning from Germany. Good morning to everyone. So a brief introduction. So AT is a rare neurodegenerative genetic disease caused by a biallelic mutation in ATM gene, and is characterized by oculocutaneous telangiectasia, cerebellar ataxia, and a high risk of developed cancer, especially leukemia and lymphoma. So the protein is ATM. It is a serine threonine kinase, and it has two main functions. It activates in double-strand break repair and in oxidative stress condition. In the past, it has been observed that glucocorticoid treatment in patients were able to lead formation of low molecular weight ATM variants, correlating with an improvement of neurological symptoms. So for this reason, my objective is develop a delivery system based on lipid nanoparticle, externally functionalized with two different scFv, and encapsulate the mRNA of these variants into this particle. This because once in the bloodstream, the scFv on lipid nanoparticle will bind the receptor on the endothelial cells, and this binding will trigger a mechanism for receptor-mediated transcytosis that will allow the particle to pass through the blood-brain barrier and fuse with neuronal cells and release the mRNA. So these are the two different scFv that we will use. At the moment, I just purify both of these scFv, and in particular, I use the anti-ICAM1 to check the functionalization of this particle. Moreover, we are also interested in study the activation mechanism of these variants. Our hypothesis is that the endogenous ATM work as a shuttle for these variants and recruit it on DNA damage site. So for this reason, what I'm doing here in Germany is using an CRISPR-Cas9 approach to eliminate the sequence here highlighted in blue. This because the sequence contains several amino acids that are necessary for ATM to interact with the mRNA complex and to recruit on DNA damage site. So this is the workflow, and I just designed to guide to target the sequence, and I just edit the XLs, and at the moment I'm working on fibroblasts. So these are the next steps, and so thank you for your attention.
Thank you, Gianluca, for your presentation. Any questions? No, there are no questions. Okay. The last PhD student of the second year is Nicois Alessandro.
Hola from Spain. Just a second that I share my screen. While I share my screen, I want to congratulate all my PhD colleagues of the 39th cycle to the awesome presentation that they've done. Okay. Do you see the presentation?
Yes.
Can you hear me well?
Yes.
Okay. Perfect. So, good evening, everybody. I'm Alessandro Nicois, and today's presentation is entitled Orexin-driven lipid droplets accumulation impairs microglial phagocytosis, evidence from in vitro and ex vivo models. Orexin is a hypothalamic neuropeptide produced by orexinergic neurons resident in the lateral hypothalamus. As we can observe from the site of production, orexinergic neurons contact many effectors in the central nervous system through two different isoforms, OX1R and OX2R. Another important actor is microglia that represent the immune compartment of the central nervous system. Microglia also maintain synaptic homeostasis, oscillating between multiple functional phenotypes. Also, microglia can undergo changes and enter specific states linked to different pathologies called disease-associated microglia. Among them, there is one characterized by an impairment of phagocytic apparatus and an accumulation of lipid droplets that is called lipid droplets disease-associated microglia. In this context, it's important to note that pathologies such as obesity, aging, and sleep-wake cycle disorders are both characterized by elevated levels of orexin A and a persistent neuroinflammation. So since microglia control neuroinflammatory processes and hyperorexemia is proven to be associated with an inflammatory state, we want to understand whether there is a link between an increase of orexin A level and microglial dysfunction. So we first analyze the accumulation of lipid droplets. Initially, we used immortalized marine BV2 cells, which were treated with orexin at a concentration of 0.3 micromolar. Exposure to the neuropeptide results in an increase of body fluorescence signal, indicating an accumulation of lipid droplets. This effect appears to be receptor-mediated, since the pretreatment with the SB 334867, these are a specific antagonist of orexin-1 receptor, prevent this effect. In the same experimental condition, we confirm the results observed in vivo cells using a primary astrocyte microglial culture obtained from CXC3R1 mice characterized by fluorescent microglia. As I said before, LDM phenotype become overloaded and thus unable to perform a proper synaptic surveillance. To verify this phenomenon, and if this phenomenon occurs in our experimental condition, we performed a phagocytosis assay, as shown in the short time-lapse video that you can see, using the Incucyte-like scanner. This video suggests that in a state of hyperorexinemia, microglia is unable to contribute properly to the environmental surveillance, and so failing to ensure a correct phagocytic behavior. As you can see, we observe a significant reduction in pH-RODO signal in cells treated with orexin A, indicating an impaired phagocytic activity. And this effect is also receptor-mediated, as the antagonist SB restore normal phagocytic behaviors. So our data demonstrate that oxate triggers lipid droplets accumulation and impairs phagocytic activity in microglial cells. And this implies that orexin-2 microglial dysfunction might play a crucial role in neuroinflammatory disease associated with LDAM. So I want to thank my supervisor, Letizia Palomba, my co-supervisors, Dr. Luigi Cristino, or the University of Urbino, and all my colleagues from CNR of Pozzuoli. And as always, thank all of you for your kind attention. If there are any questions, I would be glad to answer at the best of my knowledge. Thank you.
Okay. Well, we are a little bit tight with the time, so I hope that we can skip to the 3 minutes, so be on time. I would like to thank all the students that contributed to this first session and our moderators. Thank you very much. And we're now ready to move on to the second year PhD students presentation. So I hand you over to the moderators of this session, Elena and Alessandro. Thank you very much. And we can start.
Okay, let's start with the third year PhD students with Galvani Andrea.
So good afternoon, everybody. First of all, before jumping directly in the core of my presentation, I desire to steal you no more than a few seconds for introduce myself in a very brief way. My name is Andrea Galvani. I'm a medical doctor, and as a physician, I spent the last 13 years of work working on the emergency medicine department, sport medicine, and general practice. And I mix all these apart in one particular activity. It's still one of my activity as well, and it's the hyperbaric and diving medicine. And this is the beginning of the idea of my PhD. But before moving on, I desire to say thanks to my supervisor, Professor Pietro Gobbi, and to my co-supervisor, Professoressa Battistelli Michela. And then I can explain something about our idea. Okay. I don't want to waste time explaining what scuba diving is, because I'm quite sure that more of you know what kind of sport is the scuba diving. And you know that the most characteristic and specific chronic disease connected to this activity is the decompression sickness. And I want to underline at the beginning that basically the diving activity is quite safety because the risk of decompression sickness is very low. More or less 3 cases on the 10,000 diving. So I repeat, it's quite safe. But overall, worldwide, we see an increasing number of practitioners on the scuba diving. So as a medical doctor, we are very interested to understand better the decompression sickness. And I think that this is the most interesting part. We can split the decompression sickness in 2 different groups. The first one is called deserved, and it's occur when a diver made some mistake during the descent or during the ascent. And the second group is the undeserved decompression sickness, and they are for us very interesting because, first of all, they are more than half, 60% of the total, and at the same time, on the other end, they are interesting because you know, there is no mistakes, and the decompression sickness, when they occur, is quite dangerous for patients because there is a lot of different symptoms, and sometimes the end of these symptoms could be the death of the diver and so of the patient. So we are focused on the undeserved decompression sickness, and in the last decades we can see on PubMed and all the other paper, a lot of research work that underline the role of cardiovascular disease in the origin of undeserved decompression sickness. So we made, first of all, a research, and we saw that there is a couple of risk factors that nobody focused on the investigation. And the first one was the NAFT, so the non-alcoholic fatty liver disease, and it's worldwide recognized as a cardiovascular risk factor. So it's very easy to make the diagnosis on patient because we required only sonography. And we split our research project in 2 parts. The first part is a lab test on the cell lines to underline to understand better what happened in the cell line of liver when they are fatty. And in parallel, we made another kind of research in human using the divers and performing the sonography on the liver of this patient to see if there is some connection between the NAFT and the undeserved decompression sickness. I hope that at the end of the year we can show the end of the results, and of course I hope that we can find some interesting findings in our research. Thank you very much for your kind attention.
Are there any questions? Okay, thank you.
We move on the next one, that is Gentilini Veronica.
Good afternoon again, everyone. During the last year of my PhD project, we are analyzing the effect of exercise on the tumorigenic potential of cancer cells. To do this, we adopt cell culture models, in particular 3D cell culture models that mimic natural cancer cell growth and help us in finding relevant information to be translated into clinical practice.
In this context, exercise has been recognized for its beneficial effects in reducing cancer recurrences in cancer patients, but little is known about what type of exercises should be performed. And also the intensity, the frequency, and the duration are not clear. That's why we wanted to find some findings to know how to prevent this disease in survivors. In the first study I show you, we could demonstrate that the exercise condition serum samples obtained from breast cancer survivors that were enrolled in the MOVIS clinical trial can reduce the tumorigenic potential of breast cancer cells. Tumorigenic potential analyzed in terms of spheroid count. These women underwent a low and high intensity exercise session, and we demonstrated that especially the low intensity exercise condition serum reduces the number of spheroids. Then I went to Denmark for my abroad period, in which we did some experiments with triple negative breast cancer patient-derived organoids. In particular, we tested a set of the exercise condition serum, and we could demonstrate partially that the results were confirmed with this different 3D culture model, a little bit more complex. This is an example of the organoids stimulated with exercise condition serum. These findings suggest that for a breast cancer patient survivor, also a low intensity exercise session can be beneficial for the reduction of cancer recurrences. In the second study I present you, we analyzed the effect of exercise in terms of 2 runs, a 5 km long versus a 10 km long run. We enrolled male healthy donors that underwent these run sessions, and we collected their serum samples at rest and after exercise, and used them to stimulate 2 prostate cancer cell lines. To analyze the tumorigenic potential of these cells, we adopted a new acquired instrument called the gel count that can standardize the analysis through the detecting, counting, and also the analysis of colonies. We analyzed the number of spheroids also in this case, and we could demonstrate that the exercise condition serum, both for the 5 km long and also for the 10 km long, reduces the number of spheroids compared to the serum collected at rest. We also analyzed the total volume of spheroids, and the results highlighted that only for L and CAP cells there was a decrease in the total volume of spheroids when cultured with exercise conditions serum samples. Finally, we analyzed the determinants of spheroid count and total volume of these prostate cancer cells, and we saw that with a higher muscle mass and with a higher VO2 max, there is a lower number of spheroids and also a lower total volume in spheroids. And these findings open the way to say that when a person is more trained, maybe he can benefit more of the effects of exercise in reducing the growth of cancer cells.
we can say that this study opens the way to new in-depth analysis and also can maybe add some knowledge about the exercise that cancer survivors should practice in order to reduce cancer recurrence risk. Thank you for your attention, and thanks to my supervisor and my co-supervisor.
Thank you, Veronica. Any questions? Okay, thank you.
Okay, let's move to Tommaso Grossi.
Good afternoon, everyone. My name is Tommaso Grossi, and today I'm going to present my project entitled Testing Protocol Affects the Load Velocity Profile in Smith Machine Bench Press. The load velocity profile is based on the assumption that there is a linear relationship between load and velocity. Indeed, it is well known that when an external load is increased, we have a reduction of a lifting velocity. This loss of velocity continues until the 1 rem load is achieved. The 1 rem load is the maximal load that can be lifted in a one repetition. To date, there are no studies that have evaluated if the load velocity profile protocols can affect the slope and intercept of the linear relationship between load and velocity in different resistance exercises. Therefore, the aim of our study was to assess whether different load velocity protocol can affect the slope and intercept in the Smith machine bench press exercise by using two different execution velocity, mean velocity and mean propulsive velocity. For this reason, 17 resistant trained males were recruited. The participants performed 4 experimental sessions in a random order. Each participant, after the familiarization session, performed the Smith machine bench press exercise, and the execution velocity were recorded. The results of our study showed that the slope and intercept of the mean propulsive velocity were both affected by the load velocity protocol. Specifically, the two-point method load velocity profile produce a higher slope and intercept compared to the standardized and individualized load velocity profile. Regarding the mean velocity, only the intercept of the standardized and two-point method were significantly affected. Thank you for your attention.
We have time for one question. No, okay, thank you.
So the next one is Maestrini Sara.
So, good afternoon, everyone. I'm Sara Maestrini, and the title of my PhD project is Innovative Strategies Against Leishmaniasis, Early and Non-Invasive Diagnosis, Discovery of New Potential Biomarker and Drugs. Leishmaniasis is a complex of vector-borne parasitic disease caused by protozoa of Leishmania genus that affects large populations worldwide, including Italy, which is endemic for Leishmania infantum. Leishmania is a dimorphic parasite that develops as a promastigote in a insect vector and as a mastigote inside the host macrophages cells. In human, the disease can manifest in 3 different forms, which are the cutaneous form, the mucocutaneous form, and the visceral form, being the most serious one. There is no vaccine against leishmaniasis yet, and the therapeutic options are limited and highly toxic, showing moreover emerging drug resistance. In the last year, several studies also demonstrated how Leishmania is able to modulate the differential expression of macroRNA by host cells. And in this context, my PhD project focuses on 3 main fields of Leishmaniasis research: which are the identification of the role of microRNA in different Leishmania infection models, the drug discovery, and the development of a rapid and effective diagnostic assay using non-invasive clinical sample. Today, I will just talk about the first of these three aims of my project. And specifically, I work with three different infection models, which are human and canine macrophage-like cells infected by Leishmania infantum promastigotes, and mouse bone marrow-derived macrophages infected by Leishmania amazonensis amastigotes. The general methods includes performing in vitro infection, RNA extraction, RNA sequencing, and at the end, validation by RT-qPCR. Concerning the first two models, we also extracted RNA from extracellular vesicles secreted by cells in the culture media, and we also evaluated it-kill parasites as a control to eliminate the effect due to phagocytosis. Considering the results obtained with these two first models, we found the two most interesting macroRNA, which are macroRNA 451, found upregulated respectively in human and canine extracellular vesicle, and also macroRNA 1246, which is upregulated in human samples. But although this macroRNA is not annotated in docs, its mature sequence was identical to a fragment of a U2 spliceosomal RNA, and the qPCR assay successfully detected also this target in docs samples, suggesting that it could be potentially also a biomarker in docs. Considering the third infection models, which is the mouse bone marrow-derived macrophages infected by Leishmania amazonensis amastigotes, these results are part of my work at the Institute Pasteur of Paris, and specifically after 48 hours of infection with Leishmania amazonensis, we found 102 dysregulated macroRNA, and the qPCR for the validation was performed on these selected macroRNA, and specifically on both 5 new biological replicates of cells and infected cells, but also on ex vivo samples. And in both cases, it was possible to confirm the RNA sequencing results, and specifically the significant upregulation of macroRNA 686 and the significant downregulation of both macroRNA 99 and macroRNA 342, which have also analogs in human. I want to thank you everyone for the attention.
Thank you, Sarah. Any questions? Thank you.
Now it's the turn for Mariam Agna.
Thank you. Hello everyone, my name is Agna Mariam. I'm working with Professor Michela Battistelli on the protective effect of nutraceuticals against UVB-induced damage in human keratinocytes. So basically, UV is the high energy solar component of sun. There are 3 types of UV, UVA, B, and C. Generally, UVC is absorbed by the ozone layer, but UVA and UVB penetrates deeper into our skin. But we are working on UVB, which is the most dangerous of all, as one molecule of UVB causes 1,000 times more effect than the UVC. So these are the pathways that are generally affected by UVB. It produces reactive oxygen species, which further cause photocarcinogenesis, DNA mutation, sunburn, erythema, and it results in immune suppression. So in our previous study, we have significantly shown that curcuma caseae can protect the Hakat cells from the UVB-induced damage, and this study has extended to the live male honey. That is different from the common honey because the bees are fed on 16 types of medicinal herbs. And these 16 herbs have these properties like scavenging the reactive oxygen species, antioxidant enzyme, etc. So I will skip the hypothesis that you will know from the results. The aim of our study is first to optimize the dosage of live male honey alone, then to determine the protective efficacy of UVB, and then to characterize the cells morphologically and to analyze mitochondrial expression, and also the inflammatory cytokines. So this is the study design. First, we pre-treat the cells with the live male honey, and then we do the analysis. So these are the results we have got till now, that honey up to 100 microgram per ml is safe for the cells. But we have got two concentration that can protect the cell viability against UVB-induced damage. So these are 10 and 25 microgram per ml that shows the threshold that honey is safe and protect the cells from UVB from 10 till 25. So we have also analyzed the reactive oxygen species by DCF. So here you can see in the normal cells, the reactive oxygen species has been increased by UVB, but it has somehow reduced by pre-treating the cells with the honey, but it is not significant. But when you compare these results with the cell viability, you will see large number of cells has produced same number of DCF as compared to the small number of cells. So when you compare these results, it shows that honey significantly reduced the reactive oxygen species. Then we have also characterized the cells morphologically. Here you can see in the SEM, in the control and also in the cells that were only treated with the honey, there is a uniform surface. There is no formation of blebs, apoptotic or anything changes. But when the cells were exposed to UVB, there is increase in the formation of blebs, which were somehow reduced when the cells were pre-treated or exposed to the honey, then to the UVB. Same results were obtained at 48 hours. The cells were uniform in the control and the cells when treated with honey. But when they were exposed to the UVB, the cells were washed off. And also there was the formation of apoptotic blebs. Then we characterize the cells, check it from the TEM. Here you can see the normal morphology of the cells in both control and the cells that were only treated with the honey. There is a formation of cellular junction, there is a nucleus, nucleolus, and you can see here the intact mitochondria. It's not clear, but there are. Okay, here is also endoplasmic reticulum. So these cells, this is the normal morphology of the cells. But when the cells were exposed to the UV, then you can see there is a formation of large number of vacuoles. There is the breakdown of cellular junctions, and you can see the necrotic cells that has been thinned. But when the cells were pre-treated with honey, this morphology was somehow preserved. You can observe here, there is a cellular junction, this one, and you can also see the shape of nucleus, nucleolus, and the mitochondria there. These are, I can't see it. Okay, here, these are the mitochondria that are intact. These are the endoplasmic reticulum. So then we have also assessed the mitochondrial mass by MTG fluorescence. Here, the MTG was increased. You can see this green fluorescence. It was increased in control and also the cells that is treated with honey only. But when they were exposed to UVB, the fluorescence has reduced, but it was preserved, somehow preserved, when the cells were pre-treated with the honey. I quantified these results. Here you can see from this graph that this is the control, which shows there is the mitochondrial mass, which was reduced by UVB. But this was again to the basal line. It has come to the basal line when the cells were pre-treated with the honey. The same results were also obtained at 48 hours. Then we have checked the NRF2 localization. It can't be seen from here. It's not clear, but in the control and treated cells, the NRF2 was present in the cytoplasm, which shows the NRF2 was inactive. NRF2 is generally involved in oxidative stress pathway. When the cell is in oxidative stress, it localized from the cytoplasm to the nucleus. Here, it was moved to the nucleus. On this screen, it's not clear, but I'm telling you. But when the cells were pre-treated with the honey, the fluorescence was also observed in both nucleus and in the cytoplasm, which shows that honey somehow has preserved the cells from UVB-induced damage. So we have also evaluated the pro-inflammatory cytokines, which you can see that increased by the UV. Here. The UV has increased the inflammatory cytokines, which has somehow reduced by pre-treating the cells with the honey and then exposure to the UVB. So same results were found at 48 hours, but in case of these three molecules, it's a little different. So we have validated the results, and not till now, but we are validating these results to get the confirmed ones. So till now, you can see from the results.
Mariam, can I, if you can go faster to the...
Yeah,
okay.
I am just concluding it.
Thank you, sorry.
Okay, so till now you can see that pre-treating the cells with the keratinocyte does protect the cell morphology, mitochondrial dysfunction, ROS production, and pro-inflammatory cytokines. In future experiment, we are validating the RT-PCR experiment that I have shown, and also the Western blot and flow cytometry. Thank you for your attention. Also, my supervisor, Michaela.
Thank you. The next one is Montesi Ludovica.
Good evening, everyone. I'm Ludovica Montesi, a third-year PhD student, and today I'm presenting my project, Shear Stress and Neuropathologies, Organ-on-Chip Model to Study Neurovascular Interaction. Cerebrovascular disease are disorders that affect blood flow and blood supply to the brain. Alteration of blood flow are found in many neurological disorders, such as Alzheimer's and dementia. The neurovascular unit, a complex structure composed by multiple cell types, has a key role in regulating cerebral blood flow between the bloodstream and the central nervous system, maintaining brain homeostasis. The complexity of the neurovascular unit with ethical concern have driven to development of advanced human-relevant systems, such as organ-on-a-chip. Organ-on-a-chip are microfluidic platforms that combine in vivo and in vitro features. My PhD has three main aims: develop a human-relevant NVU organ-on-chip platform, identify the metabolic interaction in the NVU component, and target this interaction to diminish neural dysfunction. To study the effect of fluid flow on endothelial cells, I developed a 3D printed platform composed by an inlet and outlet to the connection for the peristaltic pump, and host a channel for endothelial cells. Then we perform computational simulation to study the shear stress values inside the channel. After expansing UV cells sitting in the chip, we perform calcium imaging analysis. And from the experiment, we saw a higher percentage of spiking cells and a longer duration of the spikes when the level of the fluid flow is higher. To assess the barrier integrity of endothelial cells, we create a homemade tier composed by 2 silver electrodes fixed in a 3D printed support. Our homemade tier, compared to the commercially available one, is a low-cost system and offer rapid prototyping that enable the right positioning of the electrodes in the support. To validate the system, we use 2 different cell types, CACO2 cells and UVEC cells. We culture them in transwell insert and measure the resistance through the days. Then we apply a GTA and we saw a decrease of the resistance. This was confirmed by immunofluorescence of the tight junction. In parallel, we developed another NVU organ-on-chip platform composed by 2 chambers. This platform, compared to the other platforms that are made in PDMS, avoid absorption of hydrophilic substances, making suitable for drug screening. We also perform computational simulation to study the effect of shear stress inside the chip. Then we validate the model by culturing primary neurons and UVEC cells. I had the opportunity to pass 6 months at KTH-KI involved in the Neuroenergetics on Chip project, which aims to study the metabolic interaction in the NVU component in a microfluidic platform. The microfluidic platform is composed by 3 PDMS layers that host iPSC cell-derived, the endothelial cells, pericytocyte, and the cerebral organoid. After assessing the site through profilometry assay, we perform computational modeling to see the diffusion of the species inside the microfluidic platform. After expanding the cells and sitting in the chip, we activate the flow for 48 hours, and between the different analyses, we fix the cells and we stay for the different markers of the cells. As future step in my PhD, I will further develop and expand my PhD project. Thank you for your attention.
Thank you, Ludovica.
now Sordini Enrica.
Good afternoon to everyone. I'm Enrica Sordini, and my PhD project is focused on characterize the biological activity of a new synthetic macrocycle molecule containing oxadiazole pyridine, called MOSTER, synthesized from the chemistry laboratory of Professor Fusivieri, that combines interesting biological peculiarities of oxadiazoles and macrocycles. We already assessed the antiproliferative effect of MOSTER against different human tumor cell lines. Leukemia and colon cancer are the most sensitive groups. First, we focused on pathways affected by the treatment. Transcriptomic analysis in collaboration with the European Institute of Oncology in Milan showed 58 upregulated and 142 downregulated genes affected by 24 hours of MOSTER treatment. iPathway Guide tool was used to identify the most regulated pathways, in particular mTOR signaling. In particular, mTOR signaling is the most regulated pathways involved in cellular response to stress stimuli. mTOR, in literature, there is evidence that mTOR signaling regulates autophagy, studied and proposed as a new potential anti-tumoral strategy. Proteomic analysis, in collaboration with Professor Michele Menotta, confirms transcriptomic results. mTOR signaling is the most significant pathway affected by 24 hours of MOSTER treatment. The activation of key autophagic markers was evaluated by Western blotting. In addition to molecular studies, we investigated the involvement of autophagic process by transmission electron microscopy, considered the gold standard for study autophagy, in collaboration with Professor Loretta Guidi. Figure A show abnormal not treated cells. In figure B, we observe formation of vesicles after MOSTER treatment. In figure C, we observe autophagosomes that appear as a double or multimembrane vesicles.
Excuse me, there is someone online with the microphone on. Please switch off. We can hear you. Okay, sorry, Rika.
In figure D, we observe a not treated sample in which mitochondria show their characteristic morphology. And in figure A, we observe engulfment of mitochondria by autophagic membranes, and swollen mitochondria that lost the Christie ultrastructure. Thanks for the attention, and thanks to my research team.
The next one is Sparti Angelo.
Good afternoon, everyone. I'm going to present our research on a new fully human IgG4 anti-CD99 antibody that was developed as a potential therapeutic candidate for treat acute myeloid leukemia. Why CD99? CD99 is a multifunctional protein involved in several physiological cellular processes, such as differentiation and intracellular transport. Although it is expressed normally in the most of human tissues, it's often overexpressed in solid and liquid tumors like AML. So this antibody developed by Dyativa induce selectively in AML blast and leukemic stem cells cytotoxicity, even if it has a lower toxicity in AML cell lines. An interesting feature is that this antibody cause the translocation of fleet-free ITD from Golgi to the plasma membrane, reducing its oncogenic signaling. So we are now investigating which CD99 functions are disrupted after antibody treatment and if toxic cellular dynamics is involved in this process. And this is important because we need to identify treatment-induced vulnerabilities to design dual targeting approach. That is a common approach in acute myeloid leukemia. So in leukemic stem cells from AML patients, we observed first a rapid loss of antibody bound on the plasma membrane after the treatment, and due to massive endocytosis process, and this could compromise CD99-mediated intracellular transport. We confirmed this process using two representative AML cell lines and using two different probes conjugated to antibody, one that is lysosome dependent, not shown in this slide, and another one that is pH sensitive. And we integrated the data through confocal images showing internalized complex. So importantly, this antibody shows low binding to lymphoid healthy PBMCs compared to a commercial clone targeting CD99, suggesting higher selectivity for AML blast or in general for myeloid compartment. Okay, using a surfaceome approach, that is a new strategy, promising strategy in target discovery, we found that this treatment induced strong membrane remodeling after treatment. And with the most differentially expressed membrane proteins being down-regulated after the treatment, suggesting an aberrant response of the treatment. And we are now investigating the most down-regulated protein and the few up-regulated protein, not shown in this slide, to design dual targeting approach in these days. Of course, we also assess global proteomic changes induced by antibody treatment, and in two AML cell lines, and in this slide we see only data related to MV4. And as you can see, the antibody treatment induced intracellular transport dysregulation with air stress induction, calcium dysregulation, unfolded protein response activation, and vesicle and lysosome and vesicle exocytosis dysfunction. So now we have selected some proteins that could be a direct interactor of CD99 to define a possible role in pathogenesis and in resistance. So in conclusion, we are now validating these pathways using sirna and small molecules approved by FDA or actually in clinical trial for the treatment of acute myeloid leukemia. We are also checking the transcriptional changes induced by the antibody through transcriptomic analysis and quantitative PCR. And we plan to test and to confirm this data in 20 AML patient samples that actually we have in the lab. Thank you for your attention, and a special thanks to Bianchi team and our collaborators. Thank you.
Thank you, Angelo.
Now we move to the online students, starting with Claudia Barbato. Hi, Claudia. We can see you.
Good afternoon. Today I will be talking about cervicalgia, a very common musculoskeletal condition that significantly affects both quality of life and work productivity. According to Oi et al., the annual prevalence of neck pain in the adult population ranges from 50% to 50%, with a considerable proportion of cases progressing to chronicity. This highlights the need for a comprehensive and effective treatment approach. Cervicalgia has a multifactorial etiology involving biomechanical alteration, muscular dysfunction, poor posture, and even psychological components. Cohen emphasized the importance of an accurate differential diagnosis to distinguish non-specific neck pain from secondary forms, such as radicular or discogenic pain. From a therapeutic standpoint, several studies by Hidalgo and et al. And Gross et al. Have shown that a multimodal approach is the most effective. This includes therapeutic exercise, manual therapy, postural education, in addition, stretching exercise. Stretching exercise are widely used in both acute and chronic phases to improve cervical mobility and relieve pain. A promising innovation in the field is the vibration sound system therapy. This is a neuromuscular simulation technique that uses sound frequencies applied to specific tissues. Recent clinical observation suggests that this may help reduce muscle tension and improve neuromuscular control, making it a valuable complement to conventional treatment. In conclusion, cervicalgia is a complex condition that requires an integrated approach, combining evidence-based intervention, clinical expertise, and a patient-centered perspective. This is the key to securing effective and long-term management of neck pain. Thank you.
The next one is Bartolacci Alessia.
Hi, good afternoon to everyone. One moment. Probably. Okay, yes, now I can. Can you see my presentation?
Yes, you can start.
Okay, thank you. Today, I will tell you a different story about my abroad project, and not something about my research project of gut microbiota, but something about the lung microbiota. In fact, my abroad project is about the bacterial metabolites as therapy for not small cell cancer treatment. This cancer is one of the deadliest cancer types in humans, with 20 to 30% survival rate. Microbiota dysbiosis is often associated with tumor aggressiveness. In lung cancer, preclinical models have demonstrated that lung-resident bacteria contribute to tumor aggressiveness. Until very recently, the lung tissues were considered microbe-free, but because of a constant aerosolized bacterial dispersal from the upper respiratory tract, a K-resident microbiota can be found interacting with the lungs with a metabolic crosstalk that impacts both, and it is a dynamical changing in health and disease. Recent research evidences demonstrated that K metabolites produced by specific fermentative bacteria, named short-chain fatty acids, can reprogram specific genes, reverting cancer aggressiveness in vivo. This metabolic effect can be the basis of a therapeutic approach called bacterial therapy, especially if these bacteria are lipopolysaccharide-free. I spent 3 months in Dublin and Dublin City University, where I grew 2 types of bacteria that can release a high amount of short-chain fatty acids. They are from the genera of Lactobacillus and Acidopropionibacterium, and both are microaerophilic and facultative anaerobic bacteria. This means that they can produce short-chain fatty acids in low oxygen conditions. They also metabolize specific sources of carbon, including glucose and lactose, as well as complex polysaccharides to produce the short-chain fatty acids. They are also able to adapt to the lung tissue. In fact, both were found in the lung tissue of humans and mice. Here, you can see the growth curves of the 2 bacteria. The experiments we arranged were actually flask experiments in which we grew bacteria under different conditions. It means that we had different media, such as ZMB, CIL, and DMM, and different incubation modes. You can see the growth curves of Lactobacillus and Acidopropionibacterium in different time points. We also demonstrated that all the strains could grow in DMM in 37 degrees, the typical cell culture conditions. Here we can see the HPLC results. The principal highlights are that static conditions only help release short-chain fatty acids for the first 48 hours, and Lactobacillus brevis produced 1.2 grams per liter of propionic acid in shaking condition at 50 rpm after 24 hours, and Acidopropionibacterium produced 2.6 grams per liter of acetic acid and 5.5 grams per liter of propionic acid after 192 hours in shaking condition at 70 rpm. Then I moved to Odense in Denmark to the SDU, where we are trying to create a good co-culture of human lung cancer cells A549 and bacteria, and to evaluate the epithelial to mesenchymal transition marker through Western blot. We are still in this step, finding the best settings condition for the co-culture. The next steps are to create a co-culture with the other strains grown in Dublin of Lactobacillus brevis, and with also organoids if we have time. Thank you all of you for your attention.
Thank you, Alessia. Now the next speaker is Chiara Della Franca.
Good afternoon, everyone. I share my presentation. Okay, can you see the presentation?
Yes, you can.
Okay. Good evening, everyone. Thank you for the presentation. And now I present my study, gram-negative and gram-positive bacteria-induced changes in monocyte cell population data, comparison between in vivo and ex vivo model. Firstly, I introduce sepsis. Sepsis is an uncontrolled systemic reaction to an infection characterized by the dysregulation of the immune and coagulation system, thrombosis, increased vascular permeability, tissue damages, and so on. So early diagnosis and a prompt treatment are keys to improve the outcome. Cell population data are potential useful indices of sepsis. They describe cell population by measuring the mean value and the standard deviation of volume, conductivity, and the 5 scatter of a laser light. Immune response is activated by bacteria, pathogens in general, but also by pathogen-associated molecular pattern through specific toll-like receptors, activated a cascade of events that, if exacerbated, can lead to cytokine storm, sepsis, multi-organ failure, and death. Talking about the methods of our study, we previously collected 24 blood samples in ADTK2 tubes from healthy donors treated with live bacteria, in particular with Escherichia coli 10 to 8 CFU/ml and with Staphylococcus aureus 10 to 8 CFU/ml, and their associated pump, lipopolysaccharide and lipoteichoic acid. Then we retrospectively analyze cell population data from septic patients with positive blood culture for E. coli and Staphylococcus aureus. So the aim of this study is firstly to investigate the ability of live bacteria and their pumps to modify CPD parameters, and then to compare these results, these ex vivo results, with the in vivo one, so with CPD from septic patients. So firstly, we focus, and here I show only a part of these results about monocytes, and in particular about monocytes volume heterogeneity, quantified by a cell population data, the standard deviation of volume of monocytes, from which is derived the monocyte distribution with an FDA-approved early sepsis biomarker. And here I show the absolute value results. In particular, we can see in these graphs then that both in vivo and ex vivo, there is a significant increase in this parameter, and that there are no significant differences between these two groups, while there are significant differences between the two types of control. This is due to the different anticoagulants used. So we normalize data with the default change versus control, and here we can see the first really interesting results, because we demonstrated that there are no significant differences between the three groups. So the in vivo results, so the increase in the standard deviation of volume of monocytes in patients with blood culture positive for E. coli, and compared to the same E. coli but treated in vitro, so E. coli and LPS in whole blood model. Then here we can see exactly the same results, but in the Staphylococcus aureus model. So we can see that there are no significant differences between the increase of this parameter in vivo patients affected by sepsis by Staphylococcus aureus and whole blood treated with Staphylococcus aureus and LTA. Then here I show another important results about the comparison between two groups. One about 171 patients with the sepsis by gram-negative bacteria, and the other one about 109 patients with sepsis by gram-positive bacteria. Here in the graphs, we can see that there is a significant difference between the increase in the two groups. So the standard deviation volume increase induced by gram-negative bacteria in particular is significantly higher than that induced by gram-positive bacteria. So we can conclude that firstly, the significant changes in the standard deviation of volume of monocytes induced by both live bacteria and their pumps support its important role in the early sepsis detection. Then that the study, the whole blood assay, so the model of our studies, faithfully reflects what happens in vivo under septic condition, where both E. coli and Aurus acted directly on monocytes, and that their associated pumps, so LPS and LTA, play a crucial role in this increase because of the value obtained are not different from the value obtained with live bacteria. Finally, we can conclude that there are significant differences in this parameter between the gram-negative and gram-positive bacterial sepsis, and these are results that I obtained during the first part of my period abroad in France. So I firstly thanks for my supervisor, Prof. Ferdinando Mannello, my co-supervisor, Prof. Daniel Lirici, all my team, Francesca, Marisol, and the Prof. Mark Wasser from the Hospital Foch. Thank you.
Thank you, Chiara. Now it's the time for Fondi Stephanie.
Can you see the presentation?
Yes, you can start.
Good afternoon, everyone. Today, I'll briefly introduce you to my current research in collaboration with the Neuromuscular Research Lab, which is focused on circulating extracellular vesicles as biomarkers of the stress response to US Marine Corps Mountain Warfare Training Course. Standard physical training is known to improve physiological resilience and adaptation. This resilience is vital for military readiness and sustained performance, helping the recovery from several stressors such as sleep deprivation, energy deficits, emotional strain. However, multi-stressor training can lead to chronic stress and poor adaptation. In particular, it could increase the risk of musculoskeletal injuries, which is the leading cause of lost duty time. Therefore, systemic and tissue-specific markers may help identifying resilient phenotypes. And in this context, circulating EVs may represent ideal exercise biomarker sources to monitor systemic and musculoskeletal responses to high-intensive training. For this reason, the present project aims to investigate circulating EVs released in response to the Marine Corps 6-week intensive operation to compare how their release, content, and function are altered through training and evaluate EVs as markers of resilience. Blood samples were collected from 19 participants pre, mid, and post-training and then processed to obtain plasma. EVs were isolated through size exclusion chromatography and assessed by nanoparticle tracking analysis for particle counting and size distribution, which resulted comparable to small EV size ranges reported in literature. BCA assay showed a trend in decrease in total protein levels mid-training, followed by a return near to baseline levels post-training. This is really interesting because the total protein concentration displays an opposite pattern to circulating stress marker levels, which were measured in serum, plasma, and saliva. Allostatic load index highlighted a tendency to increase during mid-training and to remain elevated compared to baseline, suggesting a possible environment of low-grade inflammation. Dot blot analysis showed that the expression of most exosome markers, such as the tetraspanin CD63, CD9, and CD81, tended to slightly decline with training, potentially indicating a suppression effect linked to cumulative acute responses from sustained high-intensity exercise. This could likely reflect a regulatory mechanism to balance intercellular communication, inflammation, and energy expenditure under chronic training stress. So altogether, these results indicate that circulating EVs could potentially reflect early molecular signaling of overtraining to predict or detect injury and therefore be used to monitor and optimize military training. In the upcoming months, we will be focused on the study of AV content through myrinomic and proteomic analysis, and finally, AV functional assays on an in vitro muscle model to characterize the role of AVs in muscle adaptation and recovery. Thank you all for your kind attention.
Thank you, Stephanie. Now it's the turn of Imperio Marta.
Yes. Okay. Can you see the presentation?
Yes, Marta?
Okay. So good afternoon to everyone from Denmark. My name is Marta Imperio. My supervisor is Francesco Lucertini. My co-supervisors are Elena Barbieri, Giosuè Balini, Fiora Guadagni, and Elvira Padua. The title of my presentation is Changes in VO2 max and triglycerides after a 12-week lifestyle intervention program may be predicted by baseline levels of inflammatory markers. So in this study, we working on data from MOVIS clinical trial, a randomized control trial, which aims to improve quality of life and cardiometabolic health of breast cancer survivors women. And we consider 27 of 138 total patients enrolled in this study. And they were randomized into different groups. Both groups received the same healthy lifestyle guidelines of physical activity and the Mediterranean diet, but only the intervention group underwent a 12-week of aerobic exercise training program with incremental intensity and duration. We assessed the cardiometabolic health, both pre and post-intervention, and the same for inflammatory markers, particularly for high sensitivity C-reactive protein, TNF-alpha, interleukin-1 beta, interleukin-6, and interleukin-10. And the main aim of this study was to verify if T0 inflammatory markers level may predict the cardiometabolic health changes observed at T1. So you can see here in this table the main cardiometabolic health changes after the intervention. For example, fat mass, insulin level, OMA index, and triglycerides improve in both groups after the intervention. And if you look at this in this line for VO2 max, for cardiorespiratory fitness level, you can see that the improvement in the intervention group was higher compared to the control group. Given these results, we decided to perform a multiple linear regression models using cardiometabolic health changes as a dependent variable and inflammatory markers as independent variables. And as you can see here for VO2 max, interleukin-6, interleukin-10, LDL cholesterol, and BMI explain 29.9% of the variance in VO2 max. And for triglycerides, interleukin-6, interleukin-1 beta, total cholesterol, OMA index, and weight explain 54.8% of the variance. And for regarding fat mass, insulin level, and OMA index, the inflammatory markers were not included into the other multiple linear regression models. So the results show us the possible role of interleukin-6, interleukin-10, and interleukin-1 beta as predictors of VO2 max and triglycerides changes in breast cancer survivor patients who underwent a lifestyle intervention program. So that's all. I would like to thank all my collaborators and our research team and all of you for the attention. Thank you.
Thank you, Marta. The next one is Piccioni, Giorgia.
Hello, everybody. Do you hear me?
Yes.
Do you see the presentation?
Yes, you can start.
Thank you. Good afternoon again. My PhD project is focusing on the role of genus Enterococcus in the spread of antibiotic resistance in humans through the food chain. As you know, Enterococci are commensal of human and animal gastrointestinal tract. However, they can act as opportunistic pathogen, especially in nosocomial area. The aim of this research.
Excuse me, Georgia.
Yeah.
Can you speak a bit louder or the microphone next to your mouth? Yes, because we can't hear you very well.
Do you hear me
Thank
well
you.
now?
Can you repeat?
Do you hear me well?
Not so much.
Now?
Better. Okay.
Okay, thank you. The aim of this research is to evaluate the actual risk of spread of antibiotic resistance in humans through the food chain, focusing on both older and more recent antibiotics, including vancomycin, erythromycin, tetracycline, and linezolid. Our strains were isolated from different sources of ready-to-eat food, including ciauscolo, raw sausages, and salami. From the molecular identification, it was found 36 strains of Enterococcus faecium that were further analyzed to know the main splitting clones using PFG. The phylogenetic tree obtained confirmed the presence of 22 different clones. The sensitivity test underlined that all strains were sensitive to vancomycin. However, 8 strains were resistant to tetracycline, 2 to linezolid, and 1 to erythromycin. The genotyping test underlined the presence of mobile antibiotic-resistant genes that could be transferred by conjugation. Then the biofilm assay showed that all resistant strains are strong biofilm producer, and this highlights the dangerousness of Enterococcus faecium. Then the conjugation test underlined the ability of resistant strains to share antibiotic resistance genes. Then we have performed a gastroduodenal digestion in vitro to evaluate its effect on resistant strain. All resistant strains survive, and their resistance profile, their ability to form biofilm, and their ability to conjugate were unchanged after digestion. Finally, we decided to perform a whole genome sequencing to have a complete view of resistant strain, and the analysis of sequencing data is currently in progress. So, in conclusion, we can affirm that ready-to-eat food acts as a reservoir of antibiotic-resistant bacteria that can reach humans through the food chain. Thank you all for your attention.
Thank you, Georgia. Now we can move to Javeira Riaz.
Can you see my presentation?
Yes, you can. Okay, perfect.
I am Javeira Riaz, a third-year PhD student, and here is my research project that I am doing at BFR in Germany. Here is the background for salmonellosis. Is the second most reported foodborne gastrointestinal infection, and I am working here on monophasic variant of Salmonella typhimurium. It is an emerging threat, and it was first identified in chicken carcass in Portugal, and it is most prevalent in Europe, United States, and Asia. And it is the third most reported from human, and it is also commonly reported from pigs and also in cattle. And it has its resistance towards ampicillin, sulfonamide, tetracycline, and along with streptomycin, it gives us R-type azote that is typical of European clone. Why it's called monophasic? Because in a normal typhimurium, there is a phase variation between FLJB and FLIC, while in monophasic variant, there is lack of the gene FLJB, or there is problem with FLJA or FLIC. Here are my objectives. We did whole genome sequencing to characterize monophasic variant isolates, and our main focus was on antimicrobial resistance genes, plasmid content, and virulence factors. And we wanted to identify the potential transmission pathways and spread of MVST in food chain in central Italy. Here is methodology. The strains were provided by Istituto Superfilatico Sperimentale from different sources, and we did serotyping, multiplex, PCR, and PFGE. Here is the whole genome sequencing methodology. We did short read Illumina and long read Oxford Nanopore technology. We used 3 bioinformatic workflows, Aquamis, Bacharach, and Chewy Snake pipelines, and Milong for hybrid assembly. Here are my results. We found that our 7 strains were positive for heme aerobie introgenic region, while all were negative for FLJB heme. So we were expecting that we might have 2 clones circulating in our food chain, and we saw into the genus, and we compared the genomic comparison. We did a genomic comparison between the heme positive and heme negative sequences, and we found that there is presence of intergenic region in heme positive, while there is no intergenic region in heme negative. In our resistance profiles, we found 11 antimicrobial classes and 29 genes, and the most important and most prevalent were ampicillin-resistant gene BLATAM and streptomycin-resistant gene APS6 and APHIB, and for sulfonamide, it was SUL2 and then tetracycline-resistant gene TETB. We also found additional resistance profiles along with azute. Here are the results of PFGE, where we found that our results correlate 100% with ONT data, and INC-Q was always found integrated into chromosome, and INC-FIB and INC-FIC were found as fusion plasmids, and there were also other INC plasmids that were found of larger sizes. What we saw in our virulence profile, there were 158 virulence genes, and they were belonging to 11 different SPIs. Overall, 75% strains carried 158 virulence genes, while the other carried 157 genes that were related to effective delivery system and adherence-related genes, as well as antimicrobial activity, nutritional factor, and immune modulation-related genes. Here are the results of the ITOL phylogenetic tree. Here we can see there are 2 distinct clusters we can see, and each cluster contain isolates from different sources, and they are showing circulation over the year in multiple settings. Here are my conclusions. Our MBST strains have the typical multi-drug resistance and even carry further resistance genes, expanding the typical azure profiles, and highly conserved and genetically stable violome. It can contribute to their transmission and persistence. Occurrence of strains from all sources in both clusters, it depicts the possibility of potential transmission of this pathogen in the food chain, and hence this study suggests that monitoring is important in line with One Health approach for public health aspect. And here are my references. And big thanks to the Department for Biological Safety at BFR and my professor, Professor Giulia Meliani and Professor Giorgio Brandi. Thanks for your attention.
Thank you very much. So we are going to present the last PhD student for this oral presentation from London, Davide Torre.
Hello, everybody. Okay, so, oh my God, desktop, that. Can you see my presentation?
Yes, Davide, you can start.
Okay, thank you.
Okay. L'abbiamo fatta.
Sorry, what? Okay, thank you very much for the presentation, and hello everybody. Today I'm going to present, sorry, I can't see properly my presentation because I have, okay. Today I'm going to present my project. In particular, I'm going to present 2 projects, the project that I performed in Urbino and the project that I will perform here in London. So the first one is on Salmonella and aim to generate a glycoconjugate vaccine, and the second one is on Klebsiella and will be the one that I will perform here. Perfect, I can't move. Sorry, guys. I need to understand how it works. Okay. Can you see the presentation?
Yes.
Okay. So Salmonella. Salmonella is an underestimated cause of invasive disease, in particular in low and middle income country. So we are trying to overcome the problem of carbohydrate-based vaccine by the generation of a glycoconjugate that, with addition of a carrier protein, aim to activate also a T-dependent B-cell response in order to generate a memory immunity and also to generate a protective immunity in children under 2 years age. And our vaccine will based on the antigen from Salmonella typhimurium for the sugar component, and the carrier protein will be the modified tetanotoxoid. So the aim of my project is to purify and characterize the two components of the glycoconjugate. Here, the result on the left, you can see the purification result for the sugar, and on the right, the purification result for the protein. And on the top, there is the quantification of the amount of the purified component at each step of purification. And we can see that for the sugar, we were able to obtain 20 milligrams of sugar, and for the protein, we obtained around 24 milligrams. So, and on the bottom of the slide, you can see on the left for the sugar, the HPLC size exclusion chromatography and the SDS page for the protein in order to evaluate the purity of the purification. And so we can conclude that the purification of the two components has been accomplished with low amount of contaminant and a total yield of 20 milligrams for the sugar and 24 for the protein. Now I'm going to talk about the project that I will perform here in England, and is based on Klebsiella, that is a high priority multi-drug resistant pathogen. And the aim of the lab is focus on the identification of new virulence factor and potential vaccine antigen. But in my particular case, my project will focus on the outer membrane vesicles composition. In particular, the aim of my project is characterize the outer membrane vesicles in terms of outer membrane protein composition and investigate the role of 6-putative autotransporter in a possible MV biogenesis involvement. So, for the first part of the project, we will isolate the OMV, and also we will analyze the protein from the outer membrane of living bacteria, and we will perform a proteomic analysis and compare the two data. And then my project will fit in a larger project that started with a bioinformatic analysis that identified the 6-putative autotransporter, and the lab generated knockout strains of Klebsiella. And here I will start my project evaluating the protein composition of the MV generated by these mutants, and comparing the result with the OMV composition from the wild type. So the future perspective in this lab is to try to define whether the OMV proteome reflect the outer membrane proteome of Klebsiella, and in the part regarding the knockout and the identified protein, we will try to understand and shed a light on the possibility that this 6 identified protein could be involved in the OMV biogenesis and composition in terms of protein. Thank you for the attention, and thank you for everybody from my Italian lab and the one from the England lab. Bye. Are you here? Yes.
Okay, thank you, Davide.
Okay,
We finish with the third-year PhD student oral presentation, so I leave the speech to Professor Manuela Frangipani.
Okay, thank you. Thank you again. I mean, I'd like to thank all the students that contributed to this session again and the moderators. Thank you very much. It is now time for the poster presentation from the first year PhD students, while we enjoy a little aperitif just outside this room. We will be back here at 7 for the award ceremony. However, since we have one PhD student from the first year who is abroad, currently abroad, he will present his poster now, and then we can leave the room and then be back at 7, okay? And the student who will present this poster now is Kai Niklas Kremers.
Hello.
Hello.
Short question, should I open now the presentation for the poster or the poster directly?
As you want, we're sharing your poster.
Okay. Okay, so you can see it?
Yes, you can start.
Okay. So yeah, I'm unfortunately abroad at my home laboratory here in Düsseldorf under the supervision of Natasha Ventura, while my co-supervisor Silvia Caloni is at your place in Urbino. And my project assesses the synergetic effect of B vitamins and omega-3 fatty acids, protecting against neuronal aging and diseases in C. elegans. So, short back to the background. So, the global increase in age-related neurological decline needs effective strategies against neurodegenerative diseases. And yeah, here diet could be a powerful tool to mitigate neurological decline. And in my project, I use C. elegans as a powerful model organism for in vivo aging and intervention studies to specifically determine the protective effect of combined supplementation of B vitamins and omega-3 fatty acids. So my aim is split into 3 objectives. The first is to determine optimal exposure scenario for both B vitamins and fatty acids. And in my preliminary results, I already have optimized B12 supplementation in different life stages with report for B12 deficiency, where I used different concentrations of B12 and different life stages of C. elegans. The second objective is to assess the impact on neuronal health by stress response behavior or disease model. It's obtained data shows Alzheimer's disease model where I applied supplementation of B12, also on different life stages, and here I see a slower paralysis of the A-beta-induced paralysis after inducing mild heat stress. And the third aim is to elucidate the underlying molecular mechanism, like mitochondrial function or metabolic changes. And here already observed high expression in antioxidant gene expression, for example, here in the GST4 after B12 supplementation. And my preliminary data indicates that vitamin B12 is non-toxic and has a beneficial effect on the model organism, but I mentioned I have to mirror this approach also for omega-3 fatty acids like DHA, and also have to apply then the synergetic effect for both diets combined. Yeah, and this was my poster presentation. Thank you all for the attention.
Thank you.
And open for questions.
Okay, thank you.
Okay, thank you very much, Kai. We can now move on and have the poster session outside while we enjoy some drinks. Thank you very much. We'll meet again here at 7.
Bye.
Piacere.